Okay, when one stem succeed in see, let me bring this one here. Okay, let's see. Okay, great. All right, so let's see. Okay, great. So when one stem, okay, when one stem succeed, okay, so let's see here. Okay, great, like that. Okay, like this one. All right. So what is going to happen is this. So when one stem, so you can see that this stem here is using its word. You see that when it gets here, all that is going to do is that it will swing. And then when it gets here, this acrosom, the tip of the acrosom, we will leave the enzyme. And this enzyme will dissolve the various layers of what? Of cells around the egg. So they will dissolve. So this is the slime membrane. Okay, so they will dissolve, and then eventually they will release this nucleus here, this yellow here, to go and fertilize the nucleus here. Okay, this one. So after one of them succeed, okay, so there's this region around the egg. We call it zona pellucida. So after one stem succeed to fertilize the egg, all the rest of the stems would die. Why? Because this zona pellucida around the egg begins to move, pushing the rest of the stems that they should go back. One has succeeded. So over 300 million stem cells, only one will win. So you sitting here listening to me, you won. That's why you are here today. Okay. So yes, so after one stem succeed in entering the egg, okay, so they will after it dissolve to release it to this nucleus and then to enter the egg and enter the small okay. So afterwards, this particular layer around the egg, which we call zona pellucida, will begin to move. Okay, so zona pellucida, and so that I can get any image of it moving backwards. Okay, right after the fertilization, to start pushing the rest of the stems that do know where go back. Somebody has come in already. Somebody has won. So I'm going to go. There's no place for you anymore. So to start moving, okay. All right. So let's see. So let's see something here. Maybe some image here like this picture. Okay. Now, so let's know what I'm. But what I'm saying here is that right after the fertilization,
Hello, Doc. Please, the screen is not showing.
Zona pellucida begins to expand forward, okay, pushing the rest of the stems here that what, enough is enough. The rest of the stem go and die because you can't fertilize the egg. One has succeeded. So it is impossible for you to get two stems can fertilize an egg. This experiment has been tried in a lot of
Please the screen.
Hello, Doc. Hello, Doc. Hello, Doc. Hello, Doc. Can you hear us? Because this part of the lectures is sweet almost everybody. Everybody wants to see the video. This is a recorded version of audio. This is the audio recording. It's not video.
But I believe you can share it. It's a video. I don't think it is. I don't think it is. How do you call it? An audio.
It's audio.
It's audio.
So it's a central actually, and this central is forming the flagellum here, and then this central here, it is believed that after the fertilization process, the zygote will undergo mitosis, a number of mitosis, okay, to forming the cleavage, the repeated mitotic division. Okay, that's the cleavage. Okay, so this particular central here helps in that process, okay, of the cell division. Okay. All right. So when they ask you question about defense, I have a question. Please go ahead. So please, my question is, what will prevent couples from not having kids or baby after going to hospital?
Yeah. Yeah, what is happening?
They are fertile.
Nothing is happening.
Compatible. Sperms are healthy.
You are waiting for dog.
Eggs are healthy. But after
because your this thing is on the recorded lectures that we had.
It's what?
Oh, wow, that's a very fun
Recorded the recorded lectures. Just like
yeah.
The question,
the
way you formulated the question is
what is it doing?
This class. Because in your question you added that everything is fine. Everything. So the things that I'm going to say, they are saying it is fine. Because if you've gotten married, they are not having baby. There are three reasons that I could say it could happen that the acrosome is not having, the enzymes of the acrosome are not really effective. Okay, the sperms don't have enough mitochondria. Okay, in the mid region, or the tail, even the tail itself, they are not strong. And sometimes we can also get something like a low sperm count. We can get something like low sperm count. Okay, and we have low sperm counts as well. All right, and so low sperm counts have also contributed to that. But your question made it this way that everything is fine. So everything is fine means that there's nothing like low sperm count. Acrosome is fine. The enzymes of the acrosomes are beautiful. Everything is okay there. The mitochondria is also there. The flagellum is also fine. Okay, because they will do all the tests to see, right? And sometimes you have some. There are some people who produce semen. They can produce semen like semen without stems. There are some people like that. Yes, so we call it azostemia. Okay, so that one they shoot glands. Okay, they shoot glands.
Hello.
There's nothing in
Hello.
so the secretions will be there.
Hello.
Just that there's no.
Hello.
Azostemia.
Please, let's lecture started.
Oh, science class, I don't believe in class.
Oh, this
No,
one is
please.
recorded. This is a recorded one. Eh?
We get me here lecture no my I think said audio been about baby.
Yeah, because I'm seeing my brother. Yeah. So I think he's rather using his audio as a revision. Or
I don't know.
okay, that's.
72 days, right? So here, so you can still look at normal days for sperm. So sometimes the frequency of sex, okay, there are some people telling they don't allow even the sperms to mature. Okay, they don't allow every day.
Yeah.
Please. And so, what you are saying now, we have a lot of, I don't know what that you call them, farm doctors or what. You see them on radio, you see them on internet saying that at least you are supposed to have this number of sex within the day. Yes, that's fine. I think. You see, for instance, your wife doesn't do every week. So, for instance, you see, hello. So, for instance, if you have sex with your wife every blessed day, okay, because you want a child, probability you waste it is very, very high because you are not waiting for the sperm to mature enough, in my opinion. Okay, so at least allow some level of sex to accumulate, like for the level of mature stem cells to accumulate very well in the world, in the epidermis. Okay, all right, but this is actually every day. That is, I'm talking about the extreme case of it. Okay, but in terms of what, I mean, if you want a child, if you want a baby, then your wife knows her ovulation.
Hello, Mr. Yaya.
She
I
means
think
that
the lecturer is in, so you meet yourself,
the ovulation.
please.
All right. Good morning. How are you?
Good morning, Doc.
Good morning, sir.
How are you
We are
doing?
Yeah, fine by His grace and you.
I'm doing well by God's grace. Okay.
we thank God for that.
Yeah. So today this is our target. In fact, we should be able to finish this because we are slightly behind. And then we'll fix just one day your reaching time, for for genetics, for the proper those are Mendelian genetic stuff. Okay, I think that one I will do it such a way that I will ask all the other groups even to join mine. Okay, because usually the SS teachers they struggle to teach Mendelian genetics, and so when you ask people what is their issue, they always tell you genetics is the problem. But genetics is one of the easiest subjects you could ever get. Okay. I think we've gotten 40 people on board, so we can kickstart now. Okay, so the last time we started talking about what? We started talking about a transport mechanism. Okay, we started talking about transport mechanism, and then from there, uh, we talked about what? We talked about let me say. Okay, so we started talking about transport mechanism. So we talked about diffusion, and then we said diffusion, movement of molecules from higher concentrated region to from a higher concentrated region to a lower concentrated region, okay, or dilute region. Okay, until a uniform mixture is obtained. Okay, until a uniform mixture is obtained. All right, so then we made mention of two types. You see, we have simple diffusion. So simple diffusion, that is a small molecules that can go through the phospholipid bilayer. Okay, so we are talking about oxygen, O2. We're talking about carbon dioxide, and we're talking about water vapor. Okay. All right, so this one can actually go. So this is by simple diffusion. All right. And then we also talked about what? We talked about another type of diffusion, which we call what? Facilitated. Another type of diffusion called facilitated diffusion. Okay, this one, it goes through what? Facilitated diffusion requires what? Requires membrane channels. Okay, channels. Okay, so that is remember the channels. Those are in cell on the cell membrane channels, right? Okay, and in this case, that is when the molecule is large. Okay, so large molecules like let's say like glucose, amino acid. All right, and then we can another one too that we can also talk about is when polar molecules. Okay, when the molecules have some sort of charge or charged, okay, molecules. So ions, okay, so like potassium ion, okay, calcium ion, sodium ions. Okay, so the reason is that if they are not lipid, um, if they are not lipid soluble, okay, they can't go through the cell membrane. Remember, we have our cell membrane here. Okay, so we have our cell membrane like this. So if you look at our cell membrane, you can see that the head region is the hydrophilic, and then the tail region is hydrophobic. So if something is not fat soluble, it cannot go through. I mean, if you are lipid, it is easy for you to go through because light dissolves light. Okay, so lipids can easily go through. Okay, so glycerol, fatty acids, and stuff like that, they can easily go through the cell membrane because they are lipid. Okay, but apart from that, large molecules like glucose and amino acid, they can't go through they can't squeeze themselves through the phospholipid bilayer. They can't go through them, so they need to be carried inside. Okay, and so they'll go through the membrane, the channels, okay, in the membrane. Okay, so the channels in the membrane, like this one, the protein channels here, that's where they will pass through. And then so those ones, large molecules, okay, large molecules like glucose, polar molecules, and then charged molecules or ions can also go through, okay, through the protein channel. All right, good. And then I made mention that facilitated diffusion. Okay, the facilitated diffusion is what? It's faster than simple diffusion. All right, and then we have one example of in the collecting that of the nephron in the kidney. Okay, let me show you a picture. Okay, let's see. All right, let me show you a picture here. Okay. So in the collection that's like that, so what happens is that in the collection that there is this special structure, we call it word. There is this special channel called the word aqua pouring. And this aqua pouring, they carry out what? Facilitated diffusion of water. They carry a facilitated diffusion of water. Okay. They carry a facilitated diffusion of water. All right, great. They carry a facilitated diffusion of water. Okay, so let me see whether I can get a clear picture of it for you here. Okay, so let me copy this and show you. Okay, so okay. Oh, why is it taking so long? It's taking so long. Okay. Oh, right. Um, I want to show you the collecting that. You know why it is taking so long like that? Then why is it taking so long? Okay. Now, if you look at this one, so this is the kidney. In the kidney, so there is this structure here. This is the kidney. There is a structure here. So this structure here is the collecting duct. That is where reabsorption of water occurs. So in this collecting duct here, okay, water absorption. But this water absorption here is under the influence of a hormone called antidiuretic hormone. So here, let's say the collecting. So cells of the collecting duct. Okay, so this collecting duct here is part of this basic structure of the kidney, which is called nephron. So this is the collecting duct here. So this collecting duct here, like that. So that's where water reabsorption will take place. So when you are very thirsty and stuff like that, there is this protein channel within this collecting duct, which we call what? We call it aquaporin. Okay, so what it does is that within this one, that is where a lot of water will be reabsorbed into the blood. Okay, so this is inside the kidney. Okay, where this nephron is located, we have this collecting duct here, where a lot of water is going to be reabsorbed. Okay, then after this one, another thing that we want to talk about is another thing we want to talk about. What are the factors that affect the rate of diffusion. What are the factors that affect the rate of diffusion? Yeah, what are the factors that affect the rate of diffusion? Yes, please.
Concentration gradient.
Good one, excellent. So one, we can talk of concentration gradient. Okay, gradients. So when you talk of concentration gradients, we are talking about, let's take it this way there. Okay, let me do some quick drawing here. So I have, let's say, I have something like this here. This is one concentration here. Okay, like that. And let's say maybe this is my membrane here. Another concentration here. I don't even need to draw it this way. So let's say this is that. Okay. So let's take it this way that Kwame is here. Let me use a simple analogy. Kwame is here. This is Kwame. Okay, let's say this is Angela. Okay, so let's say Kwame has 20,000 in his account. Let's see, Angie has 5,000 in her account. So let's say this is Angie, this is Kwame. All right, what is going to happen? So the money will move from here, okay, from higher concentrated region to go okay, until two of us will have what? A uniform mixture. until our money are the same. Let's make it, here is 25,000. Okay, 25,000. So I'll give what? I'll give 10,000 here. And when I give 10,000, here will become 15,000. Okay, and then here too will be 15,000. Then the molecules will not go again. Okay, at that point, we are what? Uniform. So look at the difference between the difference is 25,000 and 5,000. So molecules will keep moving until they all attain 15,000, 15,000. Now let's see something here. Let's take it this way, that this is Sami here, and this is Angie here again. Let's say Sami has 10,000 in her account, and then Angie still has 5,000. Okay, so you can see that, so here, Sami needs to give 2,500 here. Okay, all right, so that, and then when 2,500 move from here to here, here becomes 7,500, and here also becomes 7,500. Okay, before they will use. So you can see that if you look at the difference between Kwame and Angie, and then Angie, Sami and Angie, you can see here molecules will move faster here than the real molecules will move here, because here the concentration gradient is high. There's high concentration gradient between Kwame and what? And Angie. But here the concentration gradient is not as high as here, the one here. Why? Because if you look here, you can see clearly that here the difference is what? 25,000 and 5,000. And here is 10,000 and 5,000. Okay, so this is the analogy that I will use to explain. So because the concentration gradient, so the concentration gradient is simply the differences in what? Concentration. Okay, so it is simply the differences in what? So the differences, so when you talk of concentration gradient, okay, we are talking about the what? The difference, okay, difference in what? The differences in the concentration, okay, concentration of the two regions, all right, of the two regions. So differences, okay, so difference in the concentration of the two regions, all right, so that is what you call the word, the concentration gradient. So the differences in the word, in the concentrations of the word, of the two regions, that is the concentration gradient. Okay, great. So with this concentration gradient, okay, the steeper the concentration gradient, okay, the concentration gradient, okay, the faster the rate of what? The rate of diffusion. Okay, so here, from my analogy I gave here, you can see that the rate of diffusion between Kwame and Angie is higher than the rate of diffusion between Sami and Angie. Why? Because our concentration, the concentration gradient here is higher than the concentration gradient here. All right, great. Okay, so that is one. So now we'll talk about concentration gradient. What else again? Yeah, what other factor again?
It's a temperature.
Temperature, good. So we have the temperature. Okay, great. So can you explain to me how temperature influences diffusion in a cell? How does temperature, okay? Yeah, how does temperature influence the diffusion of what? Okay, the diffusion in cells. How does it influence diffusion in cells? Yes. Yes, okay.
So when the temperature, if the temperature is high,
yeah,
the temperature causes the molecules to exhibit kinetic energy.
Good.
High kinetic energy. And then if it is low, it slows down its kinetic energy.
Okay, that's beautiful. Okay, so temperature, we all know that temperature. So at what at a higher temperature, okay, molecules were at a high temperature, molecules move what molecules move faster. Okay, and as they move faster, which means that the rate of what, and as they do that, the rate of what, the rate of diffusion also becomes what, becomes faster or higher. Okay, so as the molecules as the temperature increases, it increases the kinetic energy, as our brother said. Okay, the Ke, okay, the Ke of what at high temperature, at high temperature, Ke, kinetic energy of the molecules. Okay, kinetic energy of the molecules what of the molecules, okay, increases, right? Okay, and that will make what, that will make diffusion increase its rate. All right, great. Which other point again? Which other point again? Yes.
It's a particle size.
Particle size, excellent. Particle size. Okay, so the size of the particles. Okay, so you can see that if the particles are in let's say powder form, okay, they are likely to diffuse faster than when they are in solid lumps. Okay, so which means that the smaller the size of the particles, okay, the faster the rate of diffusion. Okay, so particle size, okay, so smaller particle size tend to diffuse what faster. Okay, what else again? What else again? What else again?
So I want to try this one, but I don't know how to explain it. Medium viscosity. Is that also part?
When you say medium is positive, what does it mean?
As in, let's compare like the movement of honey as compared to that of gas. You see, the movement of honey is that slow, while that of the gas is a bit faster. So in terms of particles movement,
well, for the particles, once we talk of the size, I think you're talking about viscosity. It may affect it, but it's not really strong point. Okay,
okay,
the particle size is strong. Okay, once we've touched on the particle size, that's strong point. Okay, I think what if in terms of cell, what we can add is the membrane. Okay, permeability of the membrane. Permeability, if in terms of cell, okay, so let's say, let me be specific here. So in terms of cell, we can add what? Permeability of the membrane. Okay, so for instance, if you look at the like fish, if you look at the gills of fish, you see the gills of fish, the membranes are very thin. So it allows what? It allows oxygen and carbon dioxide to diffuse readily into and out of the what? Out of the blood, okay, through the gill filament, because the membranes are what? The membranes are quite thin, okay, good. So the permeability of the membrane may be in terms of what? The thickness, okay, the thickness. So you can see that thin membranes, okay, molecules can easily diffuse what? Faster through thin membranes than thick membranes. So if you look at the alveoli of our lungs, okay, if you take the alveoli, if I take something like the alveoli, alveoli, okay, alveoli of the lungs, okay, all right, of the lungs, and then the filament of the gills, okay, girls, okay, have what? Have thin, okay, okay, have thin membrane for easy word, easy diffusion, okay, diffusion of what? Of oxygen and what? Carbon dioxide. Okay, great. So in terms of cell, you can talk of the permeability of the word of the membrane, okay, its thickness. So when it is thin, okay, you can see that molecules can easily diffuse into and out of it. Okay. All right, good. And then
Doc, please, I have a question.
please go ahead.
Yes. Doc, please, let's look at the two main types of diffusion that we talked about, the
Yeah.
facilitated diffusion and then the simple diffusion. Now,
Mm-hmm.
here we are talking about the particle size. That is, the smaller the size of the particles, the faster the
Mm-hmm.
rate of diffusion.
Yeah.
Now, we are talking of the particle size in terms of when you take the simple diffusion. We made examples of water vapor, oxygen, and carbon dioxide being the
Mm-hmm.
main particles. And then when we come to the facilitated diffusion, we have glucose and the rest. Now, looking at the size of the particles of the two, you said facilitated diffusion is faster than the simple diffusion.
Mm-hmm.
Doc, please clarify something for me in terms of
It's
the
not, it's
particle
not,
size.
it's facilitated diffusion not going through channel.
Okay.
It's going through channel. It's like me going, it's like me moving from my house to UCC, okay, walking, and then walking, and then me moving from my house to UCC in a car. All right. So
Okay.
here, this one
Okay.
you are being carried. It's
Okay.
facilitated. You are being
Oh,
carried there. So once you are carried there, it wouldn't be like you going yourself. Let's take it this way that you are moving from here to Accra, okay, walking. All right, you are going on your own. Okay, and this one is like you are carried. Somebody is carrying you there. So it's like once you are carried, okay, there's a channel for you alone. So it's not that you are now going to try and squeeze yourself through. No, no, no. A channel have been opened for you alone. And the channel there is not only one. There are plenty of them that will open, okay, for you. Okay, so the moment you come, you just go through the channels, okay, and then you are going. So in that case, facilitated diffusion is faster. But what we are
okay.
saying is that the size of the molecule, even if you
Yes.
have the same molecule, and then you have it in powder form, and you have it in lump form, you can see the same molecule, but the one in powder form will diffuse faster than the one in what lump form. Okay, and if you get the one in gaseous form, if it's the same molecule, the one in gaseous form will diffuse faster than the one in powder form. So the same molecule, but the different sizes of it can even influence the rate at which they diffuse. Okay, that's what
Okay,
I
Doc.
think. All
Okay,
right.
Doc. Thank you very much.
So let's see importance of what. The next thing we're going to talk about is importance of okay, but how do you before we go to the important, how how do we demonstrate diffusion? Okay, the old school demonstration of diffusion. Let's see, creation of what diffusion. Okay, so that one, almighty potassium permanganate. You take potassium permanganate, okay, and then you drop it in what? In water. All right. So if I have something like this, and then you pick what small sample of potassium permanganate. So this is my water here, like that, like that, like that, like that, like that, like that. And then I drop a little bit of what potassium permanganate. It's purple. So I draw a little bit of potassium permanganate here. Okay, like that. So you see that in no time, you see the potassium permanganate spreading from this region of high concentration, okay, and then to spread uniformly in the way in the water. We spread uniformly in the water here. All right. Like that. So
Please, what is happening?
Log is awful. That's right.
Network.
Oh, okay, thank you. What's going on?
Hello, Mr. Yaya. I left you a message on WhatsApp, please. Mr. Yaya, I left you a message on WhatsApp, please.
Oh, okay.
Hello, my dear sister Nima too. Are you around?
Yes, please, I'm there.
I didn't get the movement yesterday.
Hello.
I said this thing you sent, it
Yes,
didn't come.
so I don't know. I have traveled to my hometown, so I have network challenges. I
Oh, okay.
sent you a lot, but as of now, it's still just downloading. I don't know why. It's still
No
loading.
problem. Come.
Yes. I'll be
No problem.
traveling back to my station, so
Hello.
it's likely to come.
Hello.
Okay. Thank you.
Hello. Please, the
Hi,
network.
sir.
Yes, the network disappeared, so I'm trying to set it up on different one. Okay. It just disappeared.
Okay.
So, all right, great. So I'm using different computer to set it up. Just okay. Sometimes MTN can be a little bit tricky. Hello, can you hear me? Hello,
Yes, sir.
can you hear me?
Yes,
Okay,
please.
okay.
Can hear you.
Okay. So, unfortunately, this one I'll find it difficult to do this thing, to draw. That's the only problem that I have with this one. Okay, let me see. Let me see. This one is coming. Okay, wait, let me see. Okay, let me try something and see. Okay. Okay, so do you know where? I think I can use, okay, so I'm print this, okay, so let me see. Okay. So can you see me? Hello, can you see me?
Yes,
Yes,
please.
please.
Yes, please.
All right, so this is better. Okay, great. I pray that this internet will not mess up again. Okay, good. All right, good. So don't worry, there's a backup now, so that in case one network misbehaves, then we can switch to another one. Okay, so so I was talking about the general diffusion stuff. Okay, so the next thing that I want us to talk about is the importance of diffusion. Okay, what are the? It's my slide. Okay, good. Okay, so okay, good. So we are talking about
Doc,
so
please.
now. Hello.
Hello, Doc, please, sir.
Hello.
Hello, please, can you hear me?
Please go ahead.
From the demonstration of
I can hear you, please.
the diffusion, from the demonstration of the diffusion
Yes, please.
method, does time affect the rate
Yes,
of
please.
diffusion?
Yes, of course. Okay, if you leave it longer, okay, what we are talking about anything that has rate. Okay, so that's what we talked about. Rate means time. Okay, so yeah, if you leave it longer, the more of them can diffuse. Okay.
Okay.
Hello. If I put a potassium permanganate and I say I'm going to stop it within 2 minutes, I'm sure maybe it wouldn't have spread through this beaker. Okay, it is, but when I allow enough time to elapse, okay, then I will see that more of them will diffuse. Okay, yeah. So time actually affects. That's why. So here the fact that we have rate here is talking about time. Okay, the rate of something. It means that we are talking about. That's why we're using the word faster. Okay, so it's affected. All right. So we are talking about. So after this demonstration of diffusion, we are talking about what are the significance of what? Significance of diffusion. Significance of a word. Significance of a word of diffusion. What are the significance of diffusion? Okay, so when you say diffusion is important, what does it mean? What are the significance of diffusion? Yeah, somebody should help me. Somebody from the audience should help me.
Okay, so one, it helps in the exchange
please,
of gases.
let's group it into plant and the plant. Okay, plant. Okay, so let's say A. Let's talk about the plants, and then after that we'll talk for animals. Okay. All right, great. Okay, so under plants, yes, let me hear from you, please. Yes, go ahead.
Okay, so under plants, I would say that it helps in the movement of nutrients.
Okay, great. So we can talk of movement of word. So movement of nutrients. Okay, nutrients. Okay, from the cell into the word, into the plant, okay, tissues via the roots, okay, through the roots, okay. All right, great. Okay, so movement, okay, so I will not limit to just move, maybe with the movement and what, and absorption of what, absorption of what, of nutrients from the soil. Okay, great. What else again? What else again? Because when I apply, let's say fertilizer, even if I apply fertilizer like, let's say NPK, if I apply fertilizer like, let's say NPK, okay, let's say fertilizer like NPK, NPK, like that, or I apply sulfate of what? Sulfate of ammonia, okay, like that. How would the fertilizer, where I apply the fertilizer is of higher concentration, and so that is going to, once it is of higher concentration, what is going to happen is going to what is going to happen is that the molecules, this NPK will move from what, higher concentrated region to lower concentrated region, okay, and so that movement or absorption of nutrients by the what, from the soil by the roots of what, plants. Okay, great. Let's see two. Yes, please.
Please, in case one applies excess amount of fertilizer in the soil, what could be the consequences to the plant?
If you apply too much of it, what could happen is that well, things, the one in the plant wouldn't come. The plant has so many defensive mechanisms in place that molecules will not move from plant to the soil. It won't happen. Okay. All right. Like there won't be reverse diffusion. It won't happen. What will happen is that it can actually poison the plant. It will be toxic. Yes, when you apply too much of it, it will be toxic. That is so. But it wouldn't be reverse diffusion. That because you've created maybe highly concentrated region here and stuff like that, or maybe the plant will accumulate more. Sometimes it will keep absorbing, and when it is too much of it, sometimes it could act as toxic to the plant. That's what will happen. But reverse of it wouldn't happen. Okay, all right. For osmosis, yes. Osmosis, yes. But the plant has so many defensive mechanisms that it will put in place. Okay, just that excess of it could poison the plant, basically. Okay.
Let me add another answer.
No.
I want to add another answer to what you said. When you apply too much fat to the crops, let's say maize or different crop, you will not get the yield. The crop will only grow. You see them nicely, but they will not produce any yield. That is another effect of it.
Okay. All right. Thank you.
Yeah. Hello.
Another explanation is that
Hello.
that one has to do with osmosis. You know, with osmosis, molecules travel from a lower concentration to a higher one. So the plant will end up wilting. It will wilt. Since the concentration in the soil or the concentration of the fertilizer is very high, water is going to move from the plant into the soil and leaving the plant to wilt.
Good. All right. Yes, somebody was also asking a question.
Yes, sir. Hello.
Yes, go ahead.
Yeah, I want to add something to what our brother said earlier. With what he said that the crop will grow, but the yield will be very difficult or it will not yield. That one is as a result of fertilizer. You have types of fertilizer, and if you want to apply it, you want the crop to yield, or you want the crop to bear food or fruit. So it depends on the one you apply. If you apply the one that you want the fertilizer to grow well, and you apply it in excess, it will let the plant grow well, but it will not bear food. But if you apply the one that the
all right,
fruit
great.
will bear,
Our focus was just on the absorption and movement. That's what we were actually focused on. We're not going into too much of the agriculture aspect. We just want to see whether the molecule will move or it will not move. Basically, that's what we're actually trying to dwell upon. Okay, so what he was asking is that when you apply more, what will happen? Yeah, so when you apply more fertilizer, yes, the plant will keep absorbing. Okay, we are not even talking about growth. All that our focus is on diffusion. What will happen at that moment? That's what we are drawing up. We are not talking about whether it will grow, the plant will grow, it will not grow. When you apply more fertilizer, okay, more fertilizer to the soil, what's going to happen? We want to know, would diffusion still okay? Yes, it will still okay. The plant will keep absorbing. Okay, so as it keep absorbing, sometimes when there is high concentration of it, if you have too much of the fertilizer in the soil, it can be toxic to the plant tissues. Okay, to some extent. All right, good. So that's also possible. And then but we are not. Our focus was not on whether the plants will grow or yield. Okay, we are just focusing on absorption or movement of nutrients. Okay, thank you. Okay, let's go to point 2. So point 2, another thing that we can also talk about when it comes to the significance of diffusion is, so we can also talk about exchange of
the gases,
carbon dioxide, okay, CO2 and oxygen, okay, through the between the atmosphere and the stomata, okay, of plant leaves. Okay, great. So you see that let me bring some sort of okay, if you remember that okay, so if you guys could remember, okay, so let me bring this diagram here for you to see. All right, if you could remember that when we're talking about the cell, okay, there was one type of word when we're talking about the cells, the plant cell. I made mention of the gut cell, okay, that the gut cell in plants, and this gut cell, what does it do? It serves as as the opening for carbon dioxide and then oxygen to enter the plant leaves. Okay, so those gut cells, okay, what they are actually using for gaseous exchange. I'm trying to get one simple image for you to see what I'm talking about. Okay, so the gut cells are for gaseous exchange. So okay, so I've gotten one image here. All right. So okay, so if you look at if you look at my structure, the structure on my screen here, okay, if you look at the structure on my screen, okay, so this is the gut cells, okay, so these two are the gut cells. So then we have the stoma is the singular, plural is the stomata. So with this stoma here, that is where the gases will pass, okay, so into the intercellular space. So we have the air spaces within here, that's the spongy mesophyll. Okay, great. So the exchange of gases between the atmosphere and the stomata of the plant leaves, okay, not only the stomata of plant leaves, you can also get it through apart from stomata of plants, you can also get through something we call lenticel of the stem. Okay, we have some who also on the stem. This place will bring about this thing. This particular space here is where the oxygen and carbon dioxide is going to enter. All right. So when the oxygen and carbon dioxide enters this place, this side, and then we'll do what we will carbon dioxide will enter here for photosynthesis, and then oxygen will exit out, okay, as a byproduct of photosynthesis. Right, good. So this particular process will occur as a result of what? Diffusion, because there's high concentration of carbon dioxide in the atmosphere than the intercellular space. And so this will cause carbon dioxide to diffuse from the atmosphere, okay, through the stomata into the intercellular space. All right, good. Now let's do that of what? Let's talk about that of all right, okay, so let's talk about that of animal cells. So yes, can you give me examples of that of animal cell? Can you give me an example for that of animal cell? Yes.
The movement of the digested food into the bloodstream.
Okay, so yeah, so movement. So you can talk of what? Absorption. So you can talk of maybe movement and what? You can talk of movement and absorption. So we can talk of movement and what? Absorption of what? Of glucose and what? And amino acid. Okay, amino acid in the word, in the microvilli. Let me make a villi of the small, okay, of the ileum. Okay, great. So we want to let's take it this way, that you want reabsorption. Okay, you want absorption. This is even just absorption. Okay, so we want the absorption. Okay, we want the absorption. All right, so here you can see that when you come to the animals, okay, we can get the word. We can get the absorption of what? Of glucose and amino acid, okay, in the villi of the small intestine, okay, into the word. Okay, so by the, okay, let me make it by, okay, by the villi of the small intestine into the bloodstream. All right, great. Into the bloodstream. All right. So we have the word. We have the movement or absorption of what? Glucose and amino acid by the villi of the word of the ileum, the small intestine, into the bloodstream. Okay, it's by the process of what? Diffusion. What else again? What else again? Yes. What else again?
Reabsorption of water in the kidney tubules.
Okay, so we can talk of reabsorption of water. So you see that in the kidney, we didn't say absorption, we say reabsorption. Okay, so let me copy this one here. Okay, so so in the kidney, we are talking about what? Reabsorption. So we can talk of absorption, not only water. Okay, so if you look at this one here, let me bring this one here. Okay, let me crop side a little bit. Okay, okay, let me bring it. Okay, so if I bring this thing here, let me duplicate my slide. Duplicate. Okay, so if you look here like this, okay, something like that. If you bring this is the nephron of the kidney. Okay, so all right, great. Now you see that kidney, in the kidney where these nephrons are located. Okay, so what is going to happen is that we have this glomerular where the blood is going to be filtered. So the ultrafiltration will occur in the glomerulus. And then we have this, you see on this side here, we have this tubule here, okay. Let me, okay, well, I don't want to go into my detail. We have this one, we call it convoluted tubule, this one here. The word convoluted means that it's folded. So this one, which is close here, this particular one, this first one, we call this one proximal convoluted tubule, okay? And then this one, this another folded one here, okay, let's see. Okay, do you know where? Let me try and bring, okay, one with, quite a very good labeling so that I show you what I'm talking about. I don't want to speak in abstract terms. Okay, good. Okay, good. So I've gotten one beautiful image here. Okay, so let's see. Then let me. Okay. All right. Now, if you look at this one, so this is more of detail of the nephron. Okay. So here, this is proximal convoluted tubule. All right. So this is. Are you there? Have I lost the link? Okay, that's fine. So this is proximal convoluted tubule. Okay. So when you come to the proximal convoluted tubule, here, so after the filtration, so we have the so ultrafiltration will occur in the glomerulus. And when it occurs that these molecules are passing here, okay, the molecules are passing here like that, like that. So when they get here, a lot of them will be reabsorbed. Okay, and this reabsorption, so this reabsorption, most of them will occur by the process of what? By the process of diffusion. Okay, because they're of high concentration. So the blood is filtered, containing the amino acid, the glucose, the nutrient, okay, and stuff like that. So they are going to reabsorb all these ones here. Okay, most of the reabsorption here will occur by the process of what? Diffusion. Remember, we are using the word reabsorption. The argument here is that the first absorption occurred in the kidney. Okay, the first absorption occurred in the kidney. All right, and then so this second absorption here is what? It's reabsorption. So this reabsorption here, okay, so the reabsorption in this place we call it what? Proximal convoluted tubule. That's where the glucose, the amino acid that is in the blood, which has been filtered going, will be reabsorbed back into the blood. Okay, and then here too, another one is going to occur, the distal convoluted tubule. Another reabsorption is going to occur there, but the vast majority of it occurs in the proximal convoluted tubule. And then, as our brother also pointed out here, and the collecting that is mainly of water. Okay, water is also reabsorbed here. In fact, actually, greater part of water is even reabsorbed here. Okay, but the reason we talk much about the collecting that is that the reabsorption of water in the collecting that is under the influence of a certain hormone. We call it what? We call the hormone antidiuretic hormone. Okay, let me crop this one a little bit so that I can have space to write. Okay, so the reason we talk much about that one is that, okay, so let's say if I bring this one here, like that, and then okay, great. So collecting that here, that's where we have the aquaporin, okay, for what? For performing, for what? For facilitated diffusion of what? Of water, okay, under the what? Under the influence, okay, under the influence of a hormone called what? ADH, antidiuretic hormone. Okay, good. All right, good. So that is how the word. That's the aquaporin in the collecting that here. Okay, so we have the aquaporins in the collecting that, okay, also carry out what? That's a reabsorption. Okay, so that's the this facilitated diffusion is the word. It's also reabsorption of what? Of water. Okay, reabsorption, okay, of water. Okay, great. So this is also here in the kidney, in the nephron of the kidney, whether the proximal convoluted tubule, the distal convoluted tubule, okay, they all reabsorb substances as a result of what? Diffusion. Okay, all right, good. All right, great. So here we can talk of a reabsorption of what? Of reabsorption of glucose, amino acid, amino acid, okay, mineral salts, okay, by the way, by the tubules of the nephron, okay, in the kidney, okay, in the kidney. All right, great. What else again? What else again that can we talk about for animals?
So we can also talk
Hello,
about the
sir.
exchange of gases in the lungs,
Okay, good. Yeah.
where oxygen moves into the lungs and then carbon dioxide exits.
Okay, that's good. So you can talk of gaseous exchange. Okay, so gaseous exchange. Okay, that's between the alveoli of the lungs. Okay, of the lungs and the atmosphere. Okay, atmosphere. So gaseous exchange, we are talking about what? We are talking about the, okay, so let me put the bracket, that's oxygen and what, CO2. Like that. All right, great. So yes, any more points? Any more points? Yes, any more points? Any more points? Yes. So gaseous exchange between the alveoli of the lungs in the atmosphere. Yes, also can occur by the process of what? Diffusion. Okay. Even gaseous exchange, yes, please.
Okay, we can also talk about the gaseous exchange. That is where there's exchange of oxygen from the lungs into the blood, and then carbon dioxide from the blood.
Okay, so we can also talk of gaseous, but it's still under gaseous exchange. Okay, so
Yeah.
here we can talk of gaseous exchange between the blood and the tissues. Okay, because when the blood transports oxygen, okay, to the tissues, when it gets to the tissues, there are more carbon dioxide in the tissues than in the blood. So that carbon dioxide from the tissue, okay, will move, okay, across the world. Okay, so through the, let me be a little bit specific here. So blood, okay, so a gaseous exchange. So gaseous exchange between the blood and the tissues through, okay, the blood through the blood capillaries. Okay, that's also nice. That's a nice point there. Okay, so between the blood, okay, so when the blood transports the oxygen and it gets to the tissues, then the oxygen will be released from the blood, to the tissues. It will diffuse across the capillary bed, okay, to the tissues, and then the carbon dioxide will also diffuse from the tissues into the blood, okay, at the capillary bed. Okay, that's also nice point. Okay, good. Any other points? I think that is, yes, please.
Can you also talk about temperature regulation?
Diffusion?
Movement of water in and out of living organisms or animals.
Temperature regulation? Not really. It's not really direct. Temperature movement is like a convection and conduction and radiation. So it's not something you can directly link to diffusion. Okay. All right. Good.
Hello, Doc.
Yes, please.
I think maybe, I don't know, but what he is trying to say must be osmoregulation instead of temperature.
Yeah, osmoregulation. See, you are saying osmoregulation. That is talking about water. Okay, which we are coming to do it very soon. Okay, great. Okay, so we've exhausted diffusion. So the next one we're going to talk about is what? We're going to talk about what we're going to talk about next is what is osmosis. Osmosis. Okay, great. So what is osmosis? What is osmosis? Yes, somebody should help me. What is osmosis? So the next, so this is what all that we've been talking about is still under what? Under passive transport. Passive.
Hello, sir.
Yes, please.
Osmosis, per old school definition, is the movement of water molecule from a region of lower concentration to a region of higher concentration.
Okay, so movement of what? Movement of water
molecule.
Water molecule here, okay, from what? From a dilute region, right? Okay,
yes, sir.
From dilute region. Okay, dilute region.
Concentrated region.
Okay, to concentrated region. Okay, it's semi
Okay, semi-permeable membrane. Permeable membrane. Great. That's beautiful definition. This definition still holds. You see that I deliberately use dilute and then I use here concentrated. Okay. I mean, yeah, what you said earlier on from a region of what lower concentration. You see, anytime we are describing concentration of solution, we describe concentration when we talk of concentration of solution, okay, we describe it in terms of solute, how much solute is there. Okay, we describe the solution in terms of how much solute. Okay, how much solute, okay, solute in the solution. That's what we describe. So anytime we are talking about concentration, we are describing the concentration of a solution, okay? We are describing about how much solute in the solution, okay? The solute, amount of solute in the word, okay? Let me put maybe the amount of what solute in the solution that describes its concentration, basically. So when we talk of what, anytime we are talking about concentration of something, we are not talking about a solvent. We are not talking about water. Okay, all right. Water is a solvent. Water is not a solute. So in the case of diffusion that we talked about previously, the one that is going to do the movement is the solute. But when it comes to diffusion, the one who is going to do the movement is the solvent, is the water. Please, do you get the difference? So actually,
yeah, please come again.
Okay, so what we are saying here is this. You see, when we're talking about diffusion, is the movement of what? We say movement of molecules. Okay, we didn't qualify the molecules. We just said movement of molecules from a region of where? From a region of high concentration to a region of low concentration. Meaning that the molecules are moving from where it is high, where its concentration is high, to where its concentration is low, or where it has high kinetic energy to where it has low kinetic energy. Good. Now you can see it is the molecule, it is the solute that is doing the movement. Okay, if I put potassium permanganate in water, you see potassium permanganate spreading. The potassium permanganate is the solute, the water is the solvent. All right, but when it comes to osmosis, you see, we say the movement of water molecule. All right, so it's the water that is moving, and water molecule is not a solute. Water molecule is a solvent. So that is where people get confused. Okay, people mess up the definition. So when we talk of osmosis, it is the water that is moving. Okay, it is not the solute that is moving. It's rather the solvent water. And because it is passive transport always means that it should move from where it is, the one who is doing the movement should move from where it is high to where it is low. So the water is actually moving from where it is high to where it is low. So when you say the osmosis is simply diffusion of water molecules. Please, do you understand? Hey. So when you say osmosis, the water molecule, it is moving from, when you say dilute solution means that solution has more water than concentrated solution. The concentrated solution has less water. So water is moving from where it is dilute to where it is what? It is more. Are we there? Hello? Okay, so let me put, so here like this. Okay, let's take this way. You see, let's take it this way like this. So we are going to prepare soakings here. Now, we decided to take the same amount of water. Okay, we'll take the same amount of water like that, the same amount of water. Okay, the cup, the same size, the same amount of water here. Okay, and then so this is Charity here. Okay, and this is Kwame here. All right, good. Now we are going to prepare solution, and you know, ladies have sweet mouth, so Charity decided to use 5 cubes of sugar. So the sugar here, 5 cubes. So Charity used what? 5. Let me use different color. Okay, so Charity used 5 cubes of sugar. Okay, all right, like that. So the cubes of sugar. Okay, okay, all right, good. And then Kwame used 3 cubes of sugar. Okay, remember the volume of water is the same. Okay, which one is dilute and which one is concentrated?
So the Charity's one is
Charity's own is concentrated. Okay, remember we took the same amount of water. So water is the same amount of water. Okay, so in terms of how much water is here, how much water is here is the same. But you see, in terms of concentration, in relation to this, you can say that Charity has what? Concentrated region, while mine is dilute. While mine is dilute region. Okay, all right. Why? Because mine contains what? More water, technically, than that of what? Than that of Charity. Even though the volume of water is the same, okay, we use the same volume of water, but Charity's own contains more sugar. So the more sugar in there, the more concentrated, okay, the solution is. So this one is of less concentration, not because the water is not enough, but it's because the solute in there. Why? Because anytime you are talking about concentration of a solution, we speak in terms of what? The amount of solute in there. Okay, when you talk of this, is the solute that we look at. The amount of solute present in the solution. Okay, so in this case, even though we took the same volume of water, okay, you can see that Charity's solution is more concentrated than mine. Okay, so if I should place a semi-permeable membrane here, if I should place semi-permeable membrane here in between Kwame and Charity, like that, and then we'll put this Charity solution and Kwame's solution here. And then you ask us that we should, so we've poured the solution, same permeable membrane here. And then you will see that the water will move from where? So you see the water will move from here, okay, to Charity's own. All right. Why? Because Charity's solution is more concentrated than mine. Let me give another scenario. Okay, let's take it this way, that in this case we'll take this cup. Okay, Kwame, if I don't want to take the same cup with Charity, let's say Charity took this cup, Kwame took this cup, Charity took this cup, okay, like this, like that, okay, and Charity, we're all giving 5 cubes of sugar to use. So this is Charity again, and this is Kwame here, all right? And then we're given 5 cubes of sugar. So look at the volume of, okay, 5 cubes of sugar, and we were asked to use all, okay? All right, good. And then, so we're all going to prepare soakings, okay? Now, Charity pick this cup and pour the word. So let's see, let's bring our sugar here. So Charity pour the 5 cubes here, and then I pour the 5 cubes here. All right, good. Okay, good. So you can see these two solutions. Which one is concentrated? You can see Charity's own is concentrated and mine is what? Dilute. Okay, even though we contain the same amount of what? We use the same amount of sugar, but you can see that the volume of water here is high. Okay, so in osmosis is simply diffusion of water molecules. So here it is the water that moves, not the solute. Okay, normal diffusion is just the movement of molecules, but here we are very specific. Is the diffusion of what? Water. Is the movement of water. Okay, and because it is passive transport, then remember it is passive transport, as I explained the last time, it doesn't require energy. And if you don't require energy, okay, if you are going to not to require energy for something, if you're not going to require energy for something, then it means that, okay, what it means here is that if you're not going to require energy, okay, it means the molecule, okay, so for
For what? For passive transport. For passive, okay, transport. Okay, transport. We know that nowhere ATP. All right. If no ATP, then the implication is that molecules should do what? Molecules should move. Molecules should move from what? From concentrated region, from conc region to dilute region. Okay. All right. So this one, what we mean is okay. So what we mean here is that okay, what we mean here is that from what? From conc region to dilute what? Region. And we say that in this case, molecules move along the concentration word, concentration gradient. Okay, so molecule, they move along the concentration gradient, which they will move from where they are high to where they are low. So when it comes to osmosis, it's the movement of water molecule. Okay, so it is not the solute that is moving, it is rather the water molecule that is moving. Okay, so you could see that when we're talking about the addition of fertilizer, there's one point that our sister gave. It was very good. I wanted to add that, but then we've not talked about diffusion. Okay, we've not talked about osmosis, so bringing it up wouldn't be the best. But I was so happy that he brought it up, so that was very, very good. So here, so if you look at this one, if you look at the passive transport, okay, so passive transport means no energy is involved. Molecules should move from where they are of high concentration to where they are of what? Low concentration. And so they move along the concentration gradient, meaning that they move from high to low. Okay, that's from what? High to low. All right, good. So in this particular case, if you look at what I just talked about right now, okay, you can see that, okay, the molecules here, the water molecules moving from where there is more water to where there is what? Less water. So you can see from here, even though all of us, we use what? We use the same amount of what? Sugar to prepare our soakings. You can see clearly that my one contains more water. Okay, because I don't want mine to be too sweet. But here is Charity. Okay, okay. So she needs hers to be sweet. Okay, so if we put Kwame here, if we put, let's say, if you put, so this is a Charity solution here and Kwame solution here. Okay, if we put the solution side by side and we put here, okay, semi-permeable membrane, all right, like that. If you put semi-permeable membrane here and then you put Kwame, so here, so which means the Kwame's own contains more water than Charity's own. True or false?
That is true.
It's true. So here, so this is dilute region. So this is Kwame's one is dilute region, and Charity's one is what? Concentrated region, like that. Okay. So dilute region means more water, more water here. Okay, more water here. All right. And then this side is what? Means less water. All right, good. So when you put this thing like that, water is going to move from a region of what? Of low concentration. But remember, low concentration means high amount of water. Okay, so for the water molecules themselves, they are not moving from where it is low. They are moving from where they are high. Are you there? But when we speak of concentration of solution, we don't speak in terms of solvent. We speak in terms of what? In terms of the solute. You see here, you are describing the concentration of these two soakings based on what? Based on the solute we added. You see here, I added 3 cubes of what? We took the same amount of water. I added 3 cubes
And then when you come to Charity pick 5 cubes. Even though the same amount of water, Charity's solution is more concentrated. Why? We are speaking in terms of the volume is the same. Volume of water is the same, but we are speaking the concentration of the soakings based on the sugar Charity added. Charity added 5, while I added what? 3. Okay, so making a solution more concentrated than mine. When it comes to the second side, now we're given 5 cubes of sugar and we're asked to use all the 5 cubes of sugar. So Charity wants sweet stuff. Okay, so because of sweet stuff, Charity pick what? This small cup. Okay, I'm going to use all the sugar as well, but I'm going to do what? I'll pick more water. So at the end of the day, you can see that this one is dilute solution. Okay, is dilute. Dilute in terms of what? The sugar, the amount of sugar and the amount of volume. If you taste mine, you see the mine is not as sweet as that of what? As that of Charity. Okay, all right. So Charity's own will be what? Will be concentrated. Okay, so when I put semi-permeable membrane here, okay, who will be doing the movement? Okay, the water will move forward. So there's more water here. So water will move from where there is less water. There is what? It will move from where there is more water to where there is what? Less water. And when anytime molecules move from where they are high to where they are low, that's diffusion. So osmosis is simply diffusion of what? Of water molecules. Please, are we clear with that? You need to get that part clearly. Okay, most of the times at JSS level, we find it very difficult to understand because we define osmosis, movements of water from low region of low concentration to a region of high concentration through semi-permeable membrane. The kids find it very difficult to relate, but you have to highlight the fact that the one who is doing the movement is not the solute, it is rather the water. And the water is moving from where it is high. Okay, dilute region of water has high amounts of water than concentrated region. So if you take my 2 solutions here, okay, the Charity solution has what? Has more solute, making it concentrated region. I have what? I have less solute, making me dilute region. But guess what? Guess who will do the movement? Is the water. So here I contain a lot of water here. Okay, I contain a lot of what? A lot of water here. So okay, more water here, more water on this side, more water here, more water here. Okay, and here less water here. Okay, like that. So when we put semi-permeable membrane here, our water will move this way. Okay, until a uniform mixture is obtained. So diffusion, osmosis is simply diffusion of water molecules. Okay, all right, from a region of what? From a dilute region to a concentrated region. And remember, dilute region means that there's high amounts of water, and concentrated region means less amounts of water. And so water will move from where it is high to where it is low. Okay, great. I hope we are clear with that. Any confusion so far? Any confusion? Okay, good. So now let's see how we demonstrate what? Let's see how we demonstrate. So let's see demonstration of what? Demonstration. Okay, so demonstration of what? Osmosis. Okay, demonstration of osmosis. Yes, good. Somebody should tell me something. Yes, demonstration of osmosis. Okay. Yes. Demonstration of osmosis. How do we demonstrate osmosis? All right. Yes. Yes.
Hello, sir.
Yes, please.
The use of yam sap.
Okay, good. So the classic one we've been doing. So we take something like a yam tuber like this. Okay, yam tuber like that. Okay, like that. Okay. All right. So then we'll scoop the middle parts of the yam. We scoop the middle parts of the yam here. All right. We scoop middle part of the yam here like this. Okay, good. And then so here, okay, we can put, what do we put? We can put here conch salt solution here. So you can put sodium chloride solution, so salt. Okay, conch salt solution here. Okay, so salt here. And we can put something like distilled water. Distilled water here. So let me put distilled water here. Okay, so this is my A. Okay, this is the experiment and this is the control. Okay, so my A here and then my B. Okay, so experiment and what? Control. All right, great. Then so I scoop this yam, then I place it in this what? In this tube, in this trough, okay, containing what? Let me use blue. Okay, then so okay, so let me, you know what? Let me use red here. So this one, okay, so this is my salt solution. Okay, so this is my salt. Okay, solution here, conch. Let me make it conch. Conch. Okay, salt solution. And then let me pick my blue here. Let me call this one distilled water. Distilled water. Okay, distilled water here. All right. And then I'll put all this one in what? In distilled water again. Like that. Okay, so I put this yam tubers here in distilled water. So this yam tuber, the yam tuber is going to act as well. The yam tuber, this tube here of yam will act as well semi-permeable membrane. Okay, to act as semi-permeable membrane. Okay, all right, to act as semi-permeable membrane. All right, great. So like that. So I will leave this yam, okay, for maybe 6 hours or maybe overnight. So I'll place it there maybe in the evening and then I will leave it overnight. Okay. So the following day, so you mark on each of them, you mark the level. So you can use a marker. Okay. So let's take it this way that I'll use maybe green marker. Okay. Let's say I use green marker to mark the level here. I mark the level here of the solution here. I mark the level of water here as well. Okay. On the yam like that. And then I will leave this setup, okay, overnight. All right. The following day, when I come, you will observe that the water level here, okay, you observe the level, okay, the level here of this one has risen. Okay, so now this one was here. Now this salt solution, you realize that it has risen, okay, up to this level, right? This level, okay, and this one has remained what? The one here has remained the same. Okay, has remained the same. So what is the explanation? So water will move. So water will move from the still water trough, okay, through the semi-permeable membrane to the what? To the source, the conch salt solution by the process of what? Osmosis. Okay, great. And then here, what will happen is that you see the distilled water here, there's also distilled water here. So the amount of water that will go here is the same amount of water that will come here. Okay, because they are of the same concentration. So the amount that will go is the same amount that will come out. All right. So at the end of the day, when you finish, you can see that the water level has risen. And the rising of the water level is demonstrating what? Osmosis, movement of what? Water from a region of what? Dilute region, okay, or a region of lower concentration to a region of what? Higher concentration. Okay, most of the time when I'm asking people to write it like that, I'll normally say if I'm going to use concentrations to define it this way: movement of water molecule from a region of what? Lower concentration of solute to a region of higher concentration of solute. That's what I normally tell my students to say it. Okay, great. Because low concentration of solute means there is more water, and higher concentration of solute means high amounts of what? Low amounts of water. Okay, so this one, the salt solution, at the end of the day, water will move from here to the what? To the tuber here, where our tuber is acting as what? As a semi-permeable membrane. It will move up here by the process of what? Of osmosis. Okay, great. So this is one way we can demonstrate osmosis. Okay, here we use a natural stuff. Okay, we can also use artificial stuff. Okay, there is this one, we call it Viskin tube. Okay, let me see that. So let's say we can also demonstrate osmosis using some sort of let me take this one off. Okay,
Okay, we can also demonstrate osmosis using Veskin tube. So Veskin tube goes this way. Okay, so okay, so let me put B here. All right, B here like this. Okay. All right, so let's say I can also demonstrate osmosis. Let me draw. So I can take Veskin tube like this. Okay, so the Veskin drawing. Okay, so we have what? So you have a tube like this, and then you have this sort of that. Okay, you turn it this way like that, and then you cover this one, this base here, with a certain membrane. So the one we normally, the membrane we call it what? Cellophane. Cellophane. That will act as what? It is acting, this cellophane is acting as what? Semi-permeable membrane. Okay, semi-permeable membrane. All right, great. And then so you prepare this one, and then you pour your Kong salt solution here, salt here, sodium chloride here, like that. And then you have and you prepare another one like that. And then this one here, the same thing like that. And then you put a cellophane here. Okay, you can actually put a Ritos stand to it. And then where you dip this one here in water. You dip this one here in water. So here, distilled water, you pour your distilled water here, and then you put all the two in what? In water. So here you put all the two in water. All right. Okay, great. Like that. Okay. Okay. I said this setup we call it what? That's the Visking tube. Okay, great. So this one we can demonstrate also osmosis using artificial medium, but remember the same permeable membrane, we can use the cellophane, okay, and stuff like that. So we leave this setup, okay, for maybe 6 hours. When you come, you can see that the water level, okay, the level of what this solution here under the source solution would have risen up here like that. So it would have gone up, and then this one remains the same, okay. So we can also use Veskin tube to demonstrate what osmosis. So you can see that the one with a higher concentration of what of solute, okay, will rise in level because water will move from the dilute region to the concentration region by the process of what osmosis. Okay. There is also another way. There is this way. Okay. Now let me show you some. You see, sometimes people underestimate people who sell in markets. Okay, our women who sell in markets. But you see, those women who sell in markets, they are smarter than we can imagine. They do what they do the science, just that they don't know the concept. They apply it all the time. Okay, just that if you ask them to explain scientifically, they cannot. Okay, but they use the concept all the time. Let me show you something here. Another way you can also demonstrate osmosis. Okay, that one you can demonstrate it with different concentration. I can choose to cut yam. Okay, yam tubers. Okay, like that. Okay, another way you can also do is that you can use potato, you can use yam. So I can use to cut yam pellets like this one. Okay, like that. Okay, this one here. Okay, 20 grams. Okay, let's say all these 20 grams. All right, these ones here, 20 grams here like this. Okay, all of them. Okay, so I decided to cut these yams. Okay, 20 grams like that. Okay, great. So all these yam pellets that I've cut here, they are of the same size. Okay, so 20 grams. Okay, 20 grams each. Okay, 20 grams each. I can use yam or I can use potatoes. Okay, so like that, 20 grams each. Then do you know what I'm gonna do? Let's take it this way, then I pick this solution. So I've picked solution here. Now look at what I'm gonna do. So I've picked solution here, normal water. So let's say this side here, I pick normal water here. Okay, water here. And then here, I pick water with salt. So let's say the concentration of salt here. So here,
is just normal water, okay? Just normal water. Here is what? It's a sodium chloride solution, but let's say the concentration, let me say it's 2 molar. Okay, let me make it 0.2. 0.2 molar concentration. Let's say this side here, another one. Okay, here, let's say I put 0.4 molar solution. 0.4 molar solution. Oh, I can't. Okay, 0.4 m. And let's say this last one here, oh, look at me. This last tube here, okay, this last one here is 0.6. 0.6 molar. Okay, so here is 0, here there's nothing in here, so 0.0. We didn't put salt solution here. Here we put salt, okay, all right, like that. And then we put this yam or potato in each of them. We put this one here in this solution, we put this one in this solution, we put this one in this solution. Okay, great. Then the following day, now let's assume that this particular yam tube has its own concentration. Let's say the concentration of salt in this one is, let's say 0. Okay, let's say 0.0. Okay, let's say 0.02. Okay, this is a concentration of what? Let's say inside the yam, the amount of sodium chloride there is 0.2. Yeah, the yam tuber or the potato tuber that I've picked. The concentration of what? Of sodium chloride there is 0.2. Okay, good. So let's see what we are going to do. All right, now I put this one here. I put this one here, put this one into this. Okay, great. Now, the following day, so let's say you put them in the evening, overnight, the following day, then you go and pick the yam, the yam tuber. So this is my yam or potato. Yes, a yam tube, yam pellets that I've cut. And you weigh this one. So you pick it and you weigh this. Do you think, would the weight increase or decrease? Would the weight, I'm asking, so would the weight, so here we know it is 20 grams. When I pick it, do you think here would the weight increase? Will it increase or it will decrease? Or it will decrease? Yes.
It will increase.
Why would it increase?
Because the water will move into the yam.
Oh, okay. Do you all believe in that? That the weight of the yam is going to increase. Okay, so here you will see that the following day when you pick this yam, okay, you can rinse it, everything, rinse the yam, make sure that you can even take a tissue paper and then wipe off the water on it and stuff like that, and put it in a beam balance or maybe a chemical balance and weigh it. When you weigh it, maybe it will come something like 2.4 grams. Maybe it will become 2.4 grams. Okay, good. Have you seen it? 2.4 grams, right? Now let's see, what about this one? What about it here? We know that the concentration is 0.2 for all of them. The yam here, 0.2, 0.2, 0.2, 0.2 here, the concentration of sodium in there. Now when I put it here, what will happen? When I put in the second one, what will happen? Yes, what will happen? Yes. Here is 0.2. This side too is 0.2. Please, what is going to happen? Yes.
So there will not be any movement of water molecule.
There's no movement of water. So what will happen to the mass? I want to know the mass.
The mass will remain the same.
The mass will
remain the same.
Remain the same. Excellent. Good. Good. So here, the mass will still be 2.0, the 20 gram. Oh, sorry. Yes, to be 24, right? I said so 24 here, sorry. Okay. So here will still be 20 grams, okay? So here will be 24 grams, here is 20 grams. All right. So here you can see that here the mass increased. Okay, here the mass remained the same. Okay, it didn't change. It stays the same. What about here? Now here it is coming into the concentration where it is what 0.4. Okay, what do you think the potato? What will happen to the yam tuber here? Yes. Yes.
But I think water molecules will move from the yam into the.
Good. So you can see that in this case, the concentration here is higher than that of the yam. So water molecules will move from that of the yam. So let's say the movement is still 2.2 points. So here, now this one becomes what? 18 grams. So which means water. So here, when you dip it here, it will rather lose water. Okay, so it will decrease. Okay, it will decrease. Okay, here remain the same. Okay, remain the same. Okay, and then this side it will remain the same because the concentration here is the same concentration here. So there's no net movement of water molecule. We shall talk about it very soon. And then here you can see that when I put the yam pellets here, it is what? It is giving us 0.4 here. So concentration here is higher than that of the yam. So water will rather move from the wet. So water, okay, water will move from the yam. Okay, water will rather move from the yam into the solution instead. Okay, water will move from the yam into the solution instead. So the following day, if we clean everything out of it, we clean everything, all the water out of it, and we weigh it, you see that the mass has come to 18 grams. Then let's see this one here. Okay, still the concentration, yes, please.
Yeah, please. The concentration down, I think is 0.1 molar, isn't it? Oh.
I said 0.2.
No, I mean the down, not the yam, the down.
Here is 0.4.
Oh, okay.
Here is 0.2, here is 4, here is 6.
Okay.
So 2,
all right.
4, 6.
Okay.
So when we come here like that, you see that there will be what? Here too is going to lose water. But here we'll lose more water. Okay, water will move more, we'll lose more water than the way here is. Why? Because the concentration difference between this and that is what? Between this and that is higher than this and that. Here the difference is 0.2 and 0.4. Difference is 0.2. Here, but the difference is what? Difference is 0.4. So here more water will be lost. So here, let's say at the end of the day, when we weigh it, we get what? 16 grams. So here too, the mass will decrease, okay, because we lose a lot of water by the process of osmosis. Okay, good. Now you see, when I started talking, I made mention of how our mothers in market, how smart they are. So for instance, if they are going to boil the yam, if they are going to boil the yam tomorrow, they are going to fry the yam tomorrow, sometimes they peel the yam and put the yam in water overnight. Okay, so they will have this big basin. They peel the yam and then put the yam in the water. They will put this yam in the water overnight. And then before they boil it, they will take it to the place where they are selling. So they will peel all the yam today. Okay. And then when they peel the yam, and then they will put it in water, okay, in big basin with water and everything. So the following day, then they will transport it to where they are going to. They will transport it to where they are going to sell their yam. And you see, when they put the yam in water, water will move into the yam by the process of osmosis. So when you finish, when they boil the yam, you see that after they cook the yam for you, Charlie, you eat the yam, 1 hour, it disappears from your body. Why? Because our mothers use the concept of osmosis, okay, to beat us. Okay, so okay, so you buy the yam over and over, you go for second round and stuff like that, they will give you this, you chop it all 30 minutes, then the yam will disappear from your body. Why? Because our mothers are smart. They are using, because when you peel the yam and you put the yam in water, and the following day, you see that the mass of the yam will increase. Okay, because water will enter the yam
by the process of osmosis. So the following day, when they fry it for you, massa, you will see that the yam is heavy. But when you chop, Charlie, 1 hour no, they will are you your family? He said, scam. Yes. Yeah, so this is scientific scamming. Okay, so they are using the concept of osmosis in a very nice way. Okay, so most of the time, if you want the yam that will use, Sati, you have to go there, they have to fry the yam right in front of you. That one is better. But
So does
say,
it have side effects?
oh, it doesn't have any side effects. It doesn't have
Oh,
side
okay.
effects.
Then
They
they
are
are
just
very scam.
there's no side. The side effect is that you will spend more because how are you?
And so will it take the same way as the original one? Will it taste nice?
It will taste nice. It will taste the same way, but more water the inside.
Then our girls, your girls, they are going to do that to you, those who are business oriented.
You see, this one I have been experiencing too. You will buy plenty of the yam, like 10 cities, but within just 30 minutes, 40 minutes time, then you feel the anger to again.
Disappear from your body.
We have been doing bad.
So Charlie, our market men, you think they don't know good. Yeah, Charlie, they know the concept of osmosis pass. Okay, all right, great. So this is also some of the simplest ways of demonstrating osmosis. Okay, so the change in mass, okay, will help us know that osmosis has taken place. Okay, so you can see that in the case of the one when the concentration of the yam and that of the solution, when the concentration was the same, there was no movement of water molecules, okay? Because no net movement. So let's say no net. Don't say no movement. So no net movement, okay, of water molecules, okay? Because the concentrations are the same. All right, good. So this is also another way of demonstrating what osmosis with different concentrations, okay? So you put the yam here, and then the following day you weigh it, okay? All right, good. This one I deliberately added different concentrations, okay? Great. All right, so that's beautiful, okay? So this is how our mothers has been scamming us low-key, okay? All right, great. So that is osmosis. So let's see what are the factors that affect the rate of osmosis. So now let's see factors that affect the rate of osmosis, okay? So factors that affect the what? That affect the what? The rate of what? Of osmosis. Okay, yes, okay.
The one.
Yes.
Concentration gradient.
Good. Concentration gradients. Okay, concentration gradients. Good. Okay, gradients. So you can see that the same explanation based on what I've done here. You can see that when the concentration gradient, the steeper the concentration gradient here, all right, at the end of the day, you can see that the water loss here is like 4 grams of water loss. Okay, all right, here, no, here is 2 grams when the concentration was 0.4. But when the concentration was 0.6, okay, which is the difference between 0.2 and 0.6 is what? It's 4. Here, the difference here is 2. 0.2, here is 0.4. So you can see that the steeper the concentration gradient, the higher the what? The rate of what? So the steeper, okay, the steeper what? The steeper the steeper the concentration gradient, the what? The higher the rate of what? Osmosis. Okay, yes, what else again? 2. Yes, any other points? Yes, 2.
So can we also talk about the membrane permeability?
Good, excellent points. Membrane permeability. Okay, membrane permeability, that's excellent point. So you can see that thin membranes, okay, so thin membranes are more permeable, okay,
than thicker membranes. Okay, all right, great. What else again? 3. Yes. 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 Factor that affecting the rate of diffusion. Yes. Yes. We talk about concentration, gradient, temperature. And temperature, they're everywhere. Temperature. Okay, temperature. And then the higher the temperature, the higher the temperature. Okay, the faster the rate of osmosis. Okay, great. What else again? What else again? You see here, you can't talk about the size of the molecules. Have you seen that you can't talk about size of molecules here? Have you seen it? Why is it you can't talk about size of molecules here?
Because with this osmosis, it is not the solid particles that move, it is a solvent.
Which solvents? Be specific.
The water molecules.
Good. So here, osmosis is exclusively to water. So here, there's nothing like different molecule or something like that. It's water. Movement of water. Okay. All right. Movement of what? Of water. Of water. So here, there's nothing like different molecules. So we can't talk about the what? The size of molecules, because the molecule is only one. It's always water that is moving. Okay, so here we can't talk about the size of the molecule. But what we can also add, what we can talk about is that we can also talk about the solubility of even the solute in there. Okay, you understand that? We can talk about the solubility of the solute in there. Okay, the solubility of the solute in the solution, okay, can also affect it. Okay, we can also talk about the solubility. Okay, so the solubility of the what? Of the solute in the solution, okay, all right, can also affect it. All right, great. So these are the factors. Now we're going to talk about the what? We're going to talk about the significance of what? Osmosis in general. Okay, so that's about significance. Okay, significance of what? Of osmosis.
Hello, sir.
Yes, please.
Yes, I want to find out something. Like the yam, there's an instance you boil one and leave one unboiled, but you put a high concentration solvent solution in the cavity of the boiled one, and then the same to the unboiled. When you leave them for some period of time, you realize that the one that is boiled, the movement will not be that as much as to the unboiled one. Can you throw more light on that?
Okay, so in your opinion, what do you think? What do you think has happened when you boil the yam?
So when you boil it, the permeability is being destroyed.
Exactly. So we've talked about. So in this case, that experiment is not demonstration of osmosis, because in this case, the two yam. You see, anytime you are conducting an experiment, okay, if I conduct an experiment, what you want to see in this case, we are demonstrating one, so every condition there should be the same. But the moment you boil one yam and the other one is unboiled, so when I boil the yam, I can't put, if I boil the one here, B here, I can't put water here. I can't put the still water here. You know what I can do? I have to put the same source solution here, because now the aim of the experiment has changed. In this case, you want to determine the permeability. Please, do you understand what I'm saying here? Because yes, now one factor has changed. Here we keep all of them living yam, but now the moment you boil one, now you've altered something. You've altered. So at the end of the day, somebody can look at our experiment and say that, oh, it's because this one is boiled. But the moment you boil this one, we can't put water here. We can't put the still water here. We have to put what? We have to put source solution here as well. Because in this case, the aim of the experiment is no longer to demonstrate osmosis, but to determine that yam is a it's a semi-permeable membrane that when it is living, it is acting as semi-permeable membrane. When it is boiled, it is not.
Not so.
Please, do you understand what I'm saying here?
Yeah, exactly. Exactly.
So that one, the aim of the experiment will change. It wouldn't be demonstration of osmosis. It will rather be determine that the experiment. I can say that one determine the experiment to determine that living yam acts as a semi-permeable membrane.
Exactly.
Okay. Yes. Okay. All right. Great. Okay. Let's see that. Hello.
Yeah. Sorry, please. The experiment you performed earlier on in a living tissue, right? Where we had high concentrated substance in the cavity of the yam and then a lower concentrated substance in the other. Yes. I wanted to find out if we can do a reverse where we put the source solution rather in the glass trough, and then we put how do we call it? Source solution.
Please, I can't see the slide.
Source solution in the glass trough. Source solution in the glass trough, and then water in one of the cavities of the yam, and then in the other one, source solution.
What do you think will happen if I put? Let's take it this way that if I put, so let's say, salt solution, conch salt solution here, conch salt here. Salt here. And then I do this.
Please, I can't see the slide.
Please, it's your network.
Okay.
It's your network, okay? Right now, I've even minimized the screen so that I can see you people. I've actually split my screen into two so that I can see you. So that when it goes off, that's one. Anytime the network goes off quickly, I detect it and then switch to different network. Okay, so here, if I have this, so let's say if I put salt concentration here, and I put this is yam here, and I put water here, and I put salt concentration here. Okay, so yeah, so if I put salt, please, that's how you describe it. If I put salt here, right?
Yes, please.
Yeah. So yeah, so osmosis will okay. It's okay this way. Water will move to here like this.
Okay.
And here, if the concentration here are the same, there won't be movement. Okay, there won't be movement. It's also osmosis was still okay. Yep.
Okay.
Okay. Okay. Good. So let's see the significance of osmosis. Yes, please.
Please, I have a question concerning the last experiment that you did concerning the market women. Hello.
Go ahead, please go ahead.
Okay. I want to find out whether it is also a recognized experiment that you can use to demonstrate osmosis, like that of the borrowed yarn sharp and that of the light skin too.
What is here, this particular one, I'm not only demonstrating osmosis, I am demonstrating osmosis with different concentration. That's the is correct. It can be used. This is osmosis demonstrating that different concentration. Okay, you use this one to demonstrate osmosis in different concentration of what of solids. Yeah, it's correct. It's also it's recognized.
And my concern is we use sodium chloride, which is recognized as table salt. Does yam really contain table salt?
I really demonstrated that we don't necessarily need to do that. If we keep asking those questions, we will know. I'll only use this one as definitely there will be even salt in there. There will be some residue of salt in there in yam. There will be sodium in there. There will be chloride in there. There will be residue of it in there. Okay. Please, the question when you are asking these are I'm explaining with analogies and stuff like that. Okay, so please, so if not, we will not move forward. Please, okay.
Okay,
These are
thank you.
analogies. Okay, so please. All
Thank
right,
you,
let's
sir.
go to significance of osmosis. Okay, so when you come to significance of what osmosis, yes, please. What are the importance of osmosis? So let's take one. Let's start with, let's say, animals. Okay, great. Let's see. Can I get one? Okay, yes, please. Significance of osmosis.
Okay,
Yes.
we can talk of preservation of fish where salt is being added to the
Uh,
fish and then put in the tank.
okay. All right. So beautiful. Okay. I get your point, but I don't want I don't want the scientific application. I don't want the scientific application. We will come to scientific application.
Oh, okay.
What you said is correct, but this is
Okay.
application of osmosis. But I want that naturally in nature, okay, like what you just said is me using the concept of osmosis for our benefit. But I want that in nature, do we have osmosis occurring in nature, okay, whereby maybe animals or cells making use of the osmosis? That's what I'm talking about. Okay, very good. Okay, so let me give you one example, like osmoregulation. Osmoregulation by the contractor, by the contractor, okay, contractor vacuum of what? Of freshwater protozoans, okay, like amoeba and what? Paramecium. Okay, amoeba and paramecium. All right, great. You see that if you take amoeba and paramecium, okay, so they have the word. If let's say if I draw something like amoeba here, let's say this is amoeba. Okay, amoeba has this one here. Now, so this amoeba is in freshwater environment, okay? And this freshwater environment, what happens is that the concentration of salt here in the amoeba is higher. So higher concentration here, and here is lower concentration. We are talking about fresh water. Okay, so which means that there is what? So more salts are here. Okay, more salts are here than salts here. All right, good. So what is going to happen is that amoeba has what? Has a contractile vacuum like this. Okay, so water will move into the cell of amoeba by the process of osmosis like this. And so when it gets into the cell, okay, it will be captured by the excess water will be captured by the contractile vacuum. Okay, so when the contractile vacuum becomes full, it will come to the surface of the cell and burst and release its contents. Okay, good. So which means that if I have two cells here, let's say I have two amoeba cells here like this. Okay, let me even make it 3 amoeba cells here. And let's say they are in fresh water. Okay, fresh water here. So let's say these 3 amoeba cells, the fresh water they are in, let's say the fresh water has a salt concentration of 0, let's say 2 point. Okay, so let's say 2.0 here. All right, let's say this is the salt concentration of 2. No, 2.0 is too big. Okay, let's say. So here salt concentration of 0. Let's say 2 here. Okay, 0.2 molar here. So this is the salt concentration here, like that. Okay. And then let's say I have 3 amoeba cells. One of them, the concentration here is 0.2 molar. Okay. Another one, the concentration here inside the cell here has is 4. Okay, 4 molar of the salt. So let's say salt. Let's say I'm using the sodium chloride. Okay, and then another one that is a concentration here is, let's say, 6 molar, like this. Now then, if I ask you the question, A, B, and C. Okay, which one would the contractile vacuole form more rapidly? Which one? So these are all amoeba cells, 3 amoeba cells. And then all of them, these 3 different amoeba cells, A, B, and C. But this amoeba cell has a concentration of salt to be 0.2. This one has 4.4. This one has 6. And then I'm asking the question, which one would the contractile vacuole form often, more rapidly? Which one? And why?
So in the 6 molar, because it will absorb
Sir, please, we cannot see from the screen. We cannot see from the screen.
Is your
really?
Please? Yep.
So the 6 molar amoeba with contractile vacuole will absorb the 0.2 molar solution from the trough into it.
To absorb what? It said to absorb what?
It will absorb the 0.2 molar.
No, it will absorb what? It will absorb what? Look at what I'm doing. It will absorb what?
Water. Is it water? Water
absorb
into the contractile vacuole. Okay.
Good.
Yes. 6 molar.
Okay. So it will. So here, there's a word that I was expecting you to say it. There's a term I was expecting you to say it. There's a term. You have to explain in terms of concentration gradient. So you can see here that the concentration gradient between this and that is huge. Okay, there's higher concentration gradient between the solution here and the cell content. So because of that, more water will be going here, and as more water gets into the 6 contractile, the amoeba with 6 molar concentration, more contractile, the contractile vacuole will capture more water, and it will be bursting quite often. Okay, great. Okay, let me give you another one. Okay, another one like this. So let's see if I give you this one here, like that. Okay, let's say now these are 2 amoeba cells. Okay, now the solution here is 2 molar. Okay, solution, and here is what? 4 molar solution. Okay, and then amoeba is 8 molar, and this amoeba is also 8 molar. Okay, and then I've put the 2 in different concentrations. Okay, so here is 2 molar, okay, all of them. So the same amoeba, I put the same amoeba, okay, but the concentration down here, the solution amoeba is in the freshwater solution here has 4, and here is 2, A and B. Which one would the contractile vacuole best more often?
So the fresh water with 4 molar.
Explain.
Due to the concentration gradient, the 4 molar, more of the fresh water enters the contractile vacuole of the amoeba in B, whereas in A there is less amount of fresh water entering the same molar of the contractile vacuole of A.
No, it's the other way around. Here the concentration is high. Here the concentration here is high. Here is 4. So if you look at the difference between them, the difference between 8 and 4 is 4.
Oh yes.
Between 8 and 6 is 4. 8 and 2 is 6. Yes. So here there's more water here than here. So in this case, okay, more water will be entering on this side. Okay, so here there will be more water. So more water, okay, because the concentration gradient between 8 and 2 is higher than what? 8 and 4. So in this case, the contractile vacuole here will be bursting more often than here. Okay, because more water will be entering the word A than in B. Okay, all right, great. Good. So this is yes, please.
Please, what is the importance of this osmoregulation in this amoeba and the parents?
Osmoregulation is control of water concentration of the cell. It's just like your kidney, what the kidney is doing in your body.
Okay, okay.
Yeah, we need to keep things in relatively stable condition. All right, so your kidney will be making sure that we have enough amounts of water in your body. The water that you have in your body, they are of like standard, okay? They should be constant, okay, so that your body can function very well. So osmoregulation, or the word osmo means water. Okay, so osmoregulation simply means controlling of the water concentration of the cell. Okay, so controlling of water concentration is very, very important because we take in more water. Like these are freshwater organisms. Water is entering their cells and they have to regulate it. Okay, so it's very, very important they regulate the amount of water in there. Okay, so osmoregulation in the contractile vacuum of what? Of fresh water occurs by the process of what? Osmosis. Yes, any other example in terms of animals? Any other example? Yes. Any other example in terms of animals? Yes. Any other example? We've talked about osmoregulation here. Any other example? Yes. Yes. Two. Which one again? Two. Yeah. Where water needs to be absorbed. Okay. Yeah. In the kidney, we just made mention of it. In the kidney. Okay. We
Hello.
can talk of what? Hello. Go
Please,
ahead.
absorption of water in the large intestine, is it by osmosis?
Absorption of water is not all that significant in the large intestine. It's not that much. Okay, like it is, but yeah, it's also go by osmosis. It's also correct. By large intestine, it's also correct, but it will go by osmosis. Okay, that's a absorption in the large intestine by the I thought you said small intestine, so that's what I said, but you said large intestine. That's correct.
Yeah, the large intestine.
You said large intestine. I thought you said initially when I heard, I thought you said small intestine. Yes. So absorption of what? Absorption. That's correct. That's excellent one. People hardly say it. You see, most of the time when people talk of absorption, is the small intestine people, but absorption of water by the word, by the cecum, okay, by the cecum of the word of the large intestine, okay, large intestine, okay, the cecum of the large intestine, okay, good, okay, it's by osmosis. Well done. That's tough one. Okay, it's by osmosis. Well done. Okay, great. Any other example? This one is tough one. Well done. Yes, next one. In the kidney. Yes. In the kidney. Okay, in the kidney. Remember, I made mention of a word. Reabsorption of a word. Reabsorption of word of water by the word, by the collecting duct. Okay, by the collecting duct, okay, of the nephron. Okay, so also okay by word osmosis. Okay, good. So reabsorption of water also occurs there. So anytime we are absorbing water, anytime water is moving, okay. So as I said, diffusion of water is osmosis. All right, good. So reabsorption of water by the word, by the collecting duct of the nephron. Okay, also occur by the process of word, by the process of osmosis. Okay, so I think I will have, and now let's see that of plants. Okay, let's see that of plants. Okay, let me duplicate this one. Let's see that of plants. Okay, good. Let's see that of plants. All right, plants. Yes, any example in plants? Examples in plants.
Absorption of water by the root hairs of
so
plants.
absorption of water, okay, by the roots, okay, by the root hairs of plants. Okay, so absorption of what? absorption of water by the root hairs of plants. That's a classic example. Okay, absorption of water by the root hair of plant. Another place that we can also talk about is you see, when the water is absorbed by the roots of plant, sometimes to the movement of water, you see, so let me draw something to show you. Okay, this is when it comes to plants. So let's see if this is the plant here. You see, you just talked about, so let's say the root hair is here. Okay, so this is the root hair. So the root hair will absorb it from the soil, right? It will absorb it from the soil like this, okay, to the plant. But when you get to the plant, let's say in here, in the middle of this plant, you have the xylem. You know, the xylem is what will transfer the water, okay, all the way from the bottom here, okay, from the bottom of the root here, all the way up to the leaves. Now, so after the water has been absorbed by the root hair to the cortex of the wood, you see, they will cross the cortex like this. So if the root plants like that, so let's say if this is the root plants like this, so here, you see, so also movements of the water here, okay. You see, after the root hair has absorbed the water here, this water should move, okay, go ahead and cross the movement through the cortex. The movement of water through the cortex, okay, all right, also occurs by the process of osmosis, okay, whereby the water moves through the cortex like this, okay, all right, so moves through the various cells in the cortex, okay. We have something we call symplastic and apoplastic pathway. We shall talk about that one in plants later on. But also the movement is not only from the shell, but also the movement of the water across the cortex to the word. So movements of words. We can also get movements of water, movements of water across the word, across the cortex to the cortex. Okay, cortex of the root, okay, to the word, to the xylem. Okay, also occurs by osmosis. Please, do you understand what I'm saying here? So you see, this is the root hair. So the root hair will absorb it first from the shell, okay, to the core. But when it comes to the cortex, it needs to move from here to the xylem before the xylem will send it up. Okay, so the movement of the water through the various cells of the cortex to the xylem, okay, also occurs by osmosis. Okay, all right, good. Okay,
Hello,
I think
sir.
Yes, please.
Hello, sir. In fact,
Go ahead.
that point, I've been saying it, but anytime I say it, then I also question myself again. Absorption of water by the root hairs of plant from the soil. We also know that in the soil, minerals that are being absorbed by the plant are dissolved in the water in the soil. So, if it is indeed absorbing water, it presupposes that that water contains the dissolved mineral particles, and the moment dissolved mineral particles appears in that water, the concentration changes. So does it mean that at a point in time, the plant absorbs water separately, and then at another time, it will absorb water dissolved with the mineral particles? Because
Okay,
they all.
That's a good question. But it's not only osmosis that plant uses to absorb. It's not only osmosis that plant uses even to absorb water. It can also use active transport sometimes.
Oh, okay.
So just like our intestines. You see, our intestine, I just said that glucose and amino acid. Okay, glucose and amino acid are absorbed by the wall of the intestine. That's the microvilli, okay, into the small intestine, into the ileum, by the process of diffusion. Okay, is that not what you said? Do you remember that's what we said, right?
Yeah.
Okay, good. This morning, most of us we've not eaten. Is that not it? Most of us we've eaten this morning.
Yes, sir.
Okay, good.
Yes, sir.
Good. We've not eaten. So the glucose content in our small intestine is lower than that of our blood now. You know, sometimes when you're even going for some tests in the lab, they will tell you that don't eat. Okay, they just want to see maybe in the morning how much sugar you have in your blood or something like that. So here, this morning, most of us we've not eaten. As we've not eaten, the glucose in our small intestine now from some of the leftover food and stuff like that that has been digested and stuff like that. Okay, that glucose concentration is what is less than that of our blood. Concentration of glucose in our blood. Have you seen it? But the glucose will not move from our blood to the small intestine. No, the cell will rather use energy and pump that small amount of glucose we have in our small intestines up, okay, against the concentration gradient into our blood. Okay, so it's not only osmosis that the plant will use. No, it's one of the mechanisms, but it will also use active transport as well. Okay, it's one of the mechanisms. Okay, it will use active transport as well. All right. If it is osmosis alone, let me tell you something. Have you seen that sometimes? Why is it that when there are some plants, when there is too much water in the soil, the plants will die? The reason the plants will die is that, you see, because it is using, sometimes plants can use active transport for absorption. That is so if there is no oxygen in the soil, if there is too much water in the soil, displacing the oxygen content, what will happen is that there will not be oxygen in the soil for plant to respire. And if there's no oxygen in the soil for plant to respire, what means that active transport requires energy. And if it requires energy, you need oxygen for you to produce energy. Remember, you need oxygen for what? For aerobic respiration, because that's what will produce a lot of energy. So if there is no oxygen in the soil, the plant root will die because it doesn't have energy that will be used to also pump the nutrients and stuff like that. Yeah, so it's not only osmosis and diffusion. Sometimes it will go by active transport mechanism, which we'll talk about it pretty soon. Okay, great. All right, great. So our sister also made mention of the previous one that when you add too much, okay, for instance, I can also kill plants by adding too much salt to the plant. Okay, so one of the old school textbooks thing of demonstrating osmosis to children is that you take earthworm and then you put earthworm in a Kong salt solution. So when you put the earthworm in a Kong salt solution, in no time you see that the earthworm is going to shrink. Okay, so you see that the earthworm is going to lose water by the process of osmosis. Okay, more water will be coming out of the earthworm and eventually it will shrink. Okay, so sometimes that is some of the if you want to demonstrate osmosis to kids in animals, we use earthworm and use very concentrated salt solution, and eventually the earthworm will lose water. And the previous question that you said, I was actually waiting to answer, but I think Nimatu or Nash or something like that explained it. So when you add too much fertilizer into the soil, that's what could also happen. Okay, when you have too much. So for instance, if I want to kill a plant, maybe in a flower pot, what I could do is that I can pour salt, okay, high concentration of salt, okay, on the plant, and that will cause what? Osmosis to move in what? In the water to move out of plants by the process of osmosis. It's a welder, okay. The whether it's a weather like it's a welder, like you see that the plants will begin to lose water, okay. So that's another thing why we shouldn't add too much fertilizer to plant. We should go by the standard dosage that has been recommended. Okay, so here for plants, for plants, we can talk about absorption of water by the root cell of plant from the soil, and you can see that the movement of water across the cortex of the root to the xylem as well also occurs by osmosis. Okay, great. Now, based on what our brother said earlier on, let's see how osmosis is applied. Okay, so that's application of osmosis. Okay, we want to see application of what? Osmosis. How is osmosis applied? Okay, so let's see application of what? Application of what? Of osmosis. Okay, osmosis. Okay, great. So in this case, that's what we are using the concept of osmosis to our benefits. Okay, how Is this one applied? Okay, great. All right, great. So let's see the application. So you realize that our brother made mention of what? Salting. Okay, good. You see that when you go to the fishing communities, you see if you go to the fish, so we use the word concept of salting. So you see before the advent of refrigeration, our mothers were using this method of preserving. Even we still use it. Okay, this method of preserving fish. You see, we have drying. The drying is where they put the fish in the sun and then to dehydrate the water. Okay, but we can also do the salting, whereby we sprinkle salt. Okay, we sprinkle what? Sprinkle salt on the what? On the fish. Okay, on the fish. We know the almighty money and stuff like that. Okay, good. Yeah, on the fish. Okay, so in this case, when we do that, what are we trying to do? When you sprinkle salt on around the fish, it will cause water. Okay, it will cause water to be drained out of the word. Okay, one, it will cause water to be drained out of the fish. Okay, so let's say this is my fish here, like that. This is my fish here, like that. Okay, like that. Okay, now if I pour salt on this fish, okay, what will happen is that I've created concentrated region around the fish. And so once I create concentrated region around the fish, it will cause water to be drained, some amounts of water to be drained out of the fish. Okay, okay, great. And then also on top of that, it will also, the bacteria that tend to cause the fish also to decay quicker. The bacteria here will also, the bacteria that will come close to it will also be dehydrated. Okay, so they will dehydrate. So when I create strong concentrated solution around the fish, it will what? It will dehydrate, to cause water to be drawn out of the fish so that it will be preserved well. And also, it also create this concentrated region around it such that when bacteria, okay, when bacteria and then other microorganisms, when they come around, they're also going to be dehydrated. Okay, because the water, they will draw water from them because of the concentrated region we've created here. And I think we also still use it in our homes. You know, sometimes when you buy the mango, okay, you want to consume it, still under this one, when you buy the mango and you want to consume it, we'll take the mango, and then we'll put it in what? In salt solution. And when you put the mango in the salt solution, what is going to happen? The mango in the salt solution is going to, in the salt solution will cause any bacteria or stuff like that that will come in, that is around the mango, to be dehydrated. So that is how come that sometimes if we don't have vinegar, we use what? Salt solution, because the salt solution is going to use the concept of osmosis to kill the bacteria that will be around. So you wash it with salt solution and you are ready to go. Okay, great. All right, another application. Okay, another application of osmosis. There's something we call reverse osmosis. Reverse osmosis. Okay, reverse osmosis is used for purification of water. Okay, used for what? It is used for purification. Okay, okay. So we can use what? You see, I think some time back we said that we are going to get water from the sea. Okay, so we can use it to obtain, to purify, purify salt water to obtain pure water. All right, okay, great. So here, but this one, it requires energy. Okay, so reverse osmosis will go this way. Okay, when it comes to reverse osmosis, what is happening is going to be that okay, we will get the osmosis like this. So here, remember, let me draw here. If this is osmosis, so strong Kong solution is here. Kong solution is here, and dilute solution is here. And then we expect water to move from where? From here. From the dilute and stuff like that to this side. Okay, great. Now, when we come to reverse osmosis, what will happen is that we will we use energy, okay? We use energy here, we apply energy here, okay, and then we'll force the what? We'll force the salt out, okay? All right, so we apply energy. So reverse osmosis actually requires energy. So it's a way of what? It's a way we use to purify seawater, okay? Let me say salt water. So it's a way we use it to purify salt water, particularly seawater. Okay, seawater. Okay, to obtain normal water from it. So that mechanism, we call it what? Reverse osmosis. So in this case, we are going to apply energy to it to get pure water out. Okay, good. So this is some of the application of what? Of osmosis in real life, where we use osmosis, okay, to preserve food substances, okay, like salting and stuff like that. So this is how we create the ammonia that we've been enjoying, okay? All right, good. And in the days of old, before refrigeration, when our great-great-grandmothers and when they bought fish, okay, that's what they were doing. They will sprinkle salt on it, and eventually they can use it, they can preserve the fish for some time. All right, good. Now, the next thing that we're going to talk about is what? It's osmosis in cells, okay? Osmosis in cells, okay? Some of them we have, okay? So let me duplicate my slide here. So the next thing we're going to talk about is osmosis. Osmosis in what? Osmosis in cells. All right, so osmosis in what? In cells, okay, great. So we're going to talk about osmosis in cells. Okay. All right, great. So let's see osmosis in cells. Now, remember that when I was talking about cells, I made mention that when I was talking about solution, okay, you see that we said solution, we describe solution in terms of the what? The amount of solute in the solution. All right, great. So we describe solution based on the word, the amount of solute. So when we say concentrated solution, what it means is that there is more, okay, so we have something like dilute solution, okay, that's a low amount of what? Solute, okay, solute. And then when we talk of what? When we talk of what? Concentrated, okay, concentrated solution, okay, when we talk of concentrated solution, we are talking about what? High amounts of what? Of solute. All right, great. So in this case like this, okay, now when we come to osmosis in cell, we are going to describe the solution in relation to the cell content. So here, let's take it this way that I have my cell here. This is my cell. Okay, so let's say this is my cell here. Now, in this case, when we buy, sometimes when you buy medicine, and you are going to, especially the syrup, if you buy it, sometimes you see that when you read it, you see tonic. It's something people, Madame Catherine tonic. There was popular one. Okay, Madame Catherine. When we were kids, they used to come around. Okay. And such like that. So you come across tonic. You come across this way like tonic. Okay, tonic here, like this. Okay, let me bring this one here. So you come across tonic. Okay. Okay, so tonic. You come across words like tonic. Tonic. Okay, great. So tonic. Okay, good. So we have something called tonicity. So the tonicity, what the tonicity does is that it compares the solution of interest with that of the cell content. Okay, so that's what tonicity is all about. So the tonicity we describe the solution. So it's what compares the solution, okay, of interest, okay, to that of the word, to that of a cell content. Okay, so that's what we use to describe. So now let's take it this way, that my cell content, okay, let's say my sodium chloride. I have sodium, let's say Sodium chloride is here. This here I have sodium chloride here. And the concentration of the sodium chloride is, let's say, 100. Let's say it's 2.0 molar concentration here. And then the concentration of what? Of sodium chloride outside the cell here, is, let's say, 0.5 molar, like this. Okay, 0.5 molar like this. In this particular case, what you realize is this, that the water, so here, okay, what you realize is this, because the concentration of salt is higher here than this one, okay, when we are describing this solution, so this solution here, okay, this dilute, this 0.5 solution, okay, so we are going to describe the tonicity of this solution. So we will say that this solution here, okay, so you can see here contain more water, okay, so you can see the solution here is of lower concentration than the cell contains. And if it's of lower concentration than the cell contains, we describe it as well as hypotonic. So when the solution is of what? So hypotonic. So hypotonic has what? Hypotonic solution has what? Concentration, that is what? That is lower than the cell contains. Lower than the cell contains, okay? Hypotonic solution has what? Has there is the solution, okay? So hypotonic solution, okay, has concentration that is of what? Lower than the cell content, okay? Then let's take it this way, that when we have, let's say, another cell, let's say we have another cell here, and the cell is like this, okay, here like that, and then this cell here has still sodium chloride to be 2.0 molar, okay, sodium chloride to be 2.0 molar, and then I have let's say here to be 4.0 molar. So in this case, you can see that the concentration of this particular cell to this one is what is high. And so when the solution is of higher concentration than the cell content, we describe it as what? Hypertonic solution. Okay, so hypertonic solution. So in this case, the concentration, okay, the concentration of the solution is higher than that of the cell content. Okay, so that is it. Okay, then whereby we have scenario whereby I have a cell, another cell like this, like that, and here is 2.0, okay, of the salt, and here is also 2.0 of the salt. So you can see that the solution here is of the same content as the solution here is of the same content as the cell. And when it happens like that, we describe that solution to be what? Isotonic. Okay, isotonic solution. And that means that the solution of what? Solution of the same concentration as the cell contains. All right, good. So you have iso, we have hyper when it is lower, hypo when it is lower. Okay, we have hyper when it is higher. Okay, when it is higher is hyper. Okay, and then when it is what? Isotonic. Okay, good. So then what we're going to do is that we are going to describe animal cell and plant cell in these solutions. Okay, good. So the first we're going to talk about is we're going to talk about we're going to talk about animal cell in what? In various solutions, okay? And we'll talk about plant cell in various solutions, okay? Good. So let's say animal cell. Let's see. So animal cell in what? In okay. So in various what? Various solutions. Okay, good. All right, and then so let's see the first one. So let's take example. I do cell, which is the red blood cell. Okay, black cell. Okay, great. So we're gonna put red blood cell in what? In different solutions, and then we'll see what we are going to get. Okay, great. Let me even delete this one here, and then let me stretch this one. Okay, like that. So let's say here, red blood cell in what? So let's say, we're going to put red blood cell in what? Hypotonic solution. Okay, in hypotonic solution. Then we'll go for another one. B, red blood cell in blood cell in hyper, okay, tonic. And then the last one is going to be, in what? C, red blood cell in what? Isotonic. Okay, tonic solution. All right. Okay, great. Okay, so let's see. Let me draw here. Now this is the red blood cell. So this is my red blood cell here, like this. Okay, like that. Okay, beautiful biconcave shape. Then I put this one in hypotonic solution. So the hypotonic solution, let's say distilled water. Okay, so yeah, so hypotonic solution here, we're talking about what? Distilled water. So you put this cell in what? In distilled water. So let's say this is hypotonic solution. This is distilled water. All right, good. What is going to happen? Yes. What is going to happen? Yes. What is going to happen? Yes. What is going to happen? Hello.
Okay. Yeah.
Hello, Doc.
Yes, please go ahead.
When the cell is in hypotonic solution, the cell will absorb water into it. And therefore, when the cell absorbs more water, it will burst.
Okay, good. Okay, so you see the answer, the way you gave your answer. In exam, I won't spoil you flow. If I should ask this question in exam and you give this, I won't spoil you flow. Yeah, you don't have to just say that it absorbs water. You have to tell us the process. Okay, so you have to tell us that. Okay, so the cell will what? Will absorb water, or water will enter the cell. Okay, by the process of what? Osmosis. Please, do you understand what I'm saying? If the word osmosis is not there, you don't get a full mark.
Okay, sir.
This is biology, not English. So you have to tell us that water will enter. So here, in this case, water will enter the red blood cell, okay, by the process of what? So here, okay, so let me, okay, so here, water will enter the what? Water will enter the what? The red blood cell by the process of what? By the process of Process of what? Process of osmosis. Okay, and remember that. So as water enter, what will happen? What's the first thing we are going to observe? The cell will what? The cell will swell. The cell.
Yes.
The cell will swell.
Will swell or increase in size.
Will swell. Okay, good. And then when it swells, when the cell swells like that, we describe it as being what? Being turgid. Okay,
yes.
Good. Flaccid is when it is drinking water. So when it goes through this thing, so the cell swells and become turgid. Okay. So in this particular case, what is okay, do you know what? Let me duplicate this my slide so that I can kick this one out. Okay, let me kick this one out here. Okay, so okay. All right, so all right, so here it will swell and become turgid. So as water enters there, the cell becomes, the red blood cell becomes bigger like this. It becomes bigger. Okay, as water enters, it becomes bigger like this by the process of what? Osmosis. So water will be entering this one by the process of osmosis like this. By the process of osmosis, water will be entering this. Okay, and as water keep entering, it will become what? It will swell and become turgid. Okay, like that. But you see, cell animal cell lack what? They lack a cell wall. So when they they don't have the cell wall. So because they don't have the cell wall, okay, because they don't have the cell wall, once after they become turgid, what are they going to do? They will burst. Okay, the cell will burst. Okay, the cell will burst. Okay, so they become like that. They will burst and then release the contents here like that. Okay, so the cell will burst and release the contents here. Okay, so here, okay, so because of what? So the cell lacks what? Lacks cell wall. Okay, it will burst after turgidity. Okay, turgidity. It will burst, okay, after turgidity. Okay, so great. So here the water will enter. It will begin to swell here, and after swelling, it will what? It lacks cell wall, and because it's lacks cell wall, it will burst and release the contents. Okay, all right, good. Now let's go to the second part. Okay, so the second part.
Please, does it mean organisms or probably let me see, organisms which have cell walls will not undergo this turgidity something?
They will become turgid by the womb burst.
Okay.
Like plant cells, they become turgid by its womb burst. All right, so let's see the second one. Let's see this. Yeah, please go ahead.
These processes occur in the blood cells. This process we are talking about here. Does it occur in the blood cells?
Yes, it's okay. Please, if you're not talking, mute. Henry, mute your mic. There's so much noise from your end.
I'm in the car.
Okay, yeah. So Henry, are you the one who asked the question?
Yes, please. Yes, please. I'm the one.
So yeah. So it occurs in our it occurs in our cells. Okay, it occurs there as well. Okay, it occurs there. So I will the last part. I will add maybe something that could it is very important clinically. So I will add it very soon. Okay. Yes, another person is also asking a question. Please go ahead. Hello. Yes. Another person was asking a question. Okay. All right. So when you come to the hypertonic solution, so we have when you come to hypertonic solution, that's when you put the cell. So let's say the red blood cell. Okay. So let's take this one. Let me take this So I'll put the red blood cell in what? So let's say this one here. Okay. So this is the solution. Okay, and then like that. Okay, so here let's assume that the content is let's say let me give a content like, okay, so let's say let me say concentric word salt solution. All right, salt word. So ACL, concentrated ACL here, concentrated ACL, okay, like that here. So now the solution is what? So here the cell has been placed in what? So you can see now the cell has been placed in hypertonic what? Hypertonic solution. Okay. So in hypertonic solution, so we are using what? ACL here. So ACL means that the cell content is what? So cell red blood cell in hypertonic solution. Okay. So let's say what? Concentrated with ACL, like that. So what is going to happen is this. In this case, water will begin to move from from here, from the cell to the source solution this way. Water will begin to move from the cell to the source solution like this, like that. Okay, good. And so when this one happens, what is going to happen? Let's see. Okay, so water to begin to lose water. Okay, so water will move where? So here, water will move out of the word. Will move out of the cell by the process of what? Osmosis. Okay, osmosis. Okay, good. And then as water keeps moving out of the cell, what is going to happen? The living component, okay, so the cell will do it. So here, if I should draw again, so it's beginning to work. It becomes what? So this is where it becomes good. This way it becomes flaccid. Okay, flaccid. Okay, so what? So it loses water and it becomes what? Flaccid. And then after the flaccidity, okay, so and then it will what? It will shrink. Okay, good. So let me do some drawing here. So here, so as the cell keeps losing water, so it is becoming, so this our nice cell is losing water. Okay, every style be happy. And so as it loses water like that, okay, it's losing water from all angles like this. Okay, like that. So as it loses the water, it is shrinking. So this shrinking, okay, this nature is the flaccid. Okay, it has become flaccid here. Okay, flaccid. Okay, it has become flaccid. And then the final stage, okay, it will shrink completely. It will shrink like that. Okay, and then the shrinking of animal cell, what's the term for it? Yes, what's the term for the shrinking of animal cell? With the shrinking, okay, of the protoplasm of the
So is that plasmolysis?
That is for plants. Plasmolysis is for plants. This is correct.
Okay.
For plants, okay.
Placidity.
Okay, we call it shrinking of the cell of animal cell of the animal cell.
Plasmolysis.
Okay, when placed in a solution that is higher than its content, higher in concentration, okay, than its content. Okay, we call it
plasmolysis.
No, it's called cremation.
Cremation. Okay.
This is the right term. We hardly hear of it as a, but that's a cremation. Okay.
Okay. Thank
That's
you.
a cremation. That's the right word. Cremation. Okay, that's a cremation. Okay, great. All right, great. So let me get you simple images that okay, so let me get you simple images that you can actually, okay, all right, okay, so let's see, so that, okay, so let's see, okay, so cells in, okay, so let's see, in hypo, okay, good, so let me see whether I could get some simple images for you, okay, so that you have idea of what I'm talking about, okay, so that, okay, great, so let's see something like this, okay, let me see something like that, okay, great, okay, so let me copy this one for you, all right, great, okay, all right, so let's see, let me, so let me enter the one I just did. Okay, good. Okay, good. So let's say, let me put this one for you here. Oh, this image is too small, but I think you can see what I want you to see. Okay, good. So here like that. Okay, good. So the red blood cell here. Okay, good. So this, when you put it in hypertonic, it will shrink, and when it shrinks, it's called what? That's a crenation. That's a technical word. Okay, we use the plasmolysis for plant and crenation for animal cell. Okay, so it's called what? Crenation. What I wrote. Crenation. Okay, good. So this one. Okay, good. All right, good. So we have the crenation. And look at the pictures here. Okay, great. Then I'm going to ask you a question. Okay. The question I'm going to ask you is this. A nurse was in a hurry to go home. Okay, a nurse. Okay, but maybe before I ask that question, let me do the last one before I ask that question. So let's say this is that, so you can see this is the hypertonic solution. The cell is going to shrink, and the shrinking of the animal cell is called crenation. When you put it in hypotonic solution, it will absorb water, it will swell, and it will burst. Okay, so here, from here, it will lose water, becomes weak, which is called flaccid. After flaccidity, it will shrink. And the shrinking of the living component or the protoplasm of the cell, of the animal cell, is called crenation. Okay, crenation. And then the best thing here is after tegility, there's no cell wall, it's going to burst. Okay, good. And let's see that of the isotonic. So let me move all that. And then, okay, so let's take isotonic solution. So let's say isotonic solution, all right, like that. Okay, you put the cell beautifully in isotonic solution, like that. Okay, so when you put it in isotonic solution, so here, if here is 2 molar, the down here is also 2 molar. Okay, good. In isotonic solution, please, what is going to happen? Yes, so what will happen? Okay, yes, what will happen? When the cell, yes, please.
The movement of water in and out of the cell through osmosis will be 0.
Okay, good.
So therefore, the cell will maintain its shape.
Okay, great. So here, the term you need to use is there is what? We use the term no net movement. Okay, when you say no net, what it means, no net movement of what? Of water molecules. What it means here is that what goes is the same as what comes out. Okay, so sometimes if you want to trick the people, we just say that there's no water movement. No, if you say that, that's incorrect. There's water movement, but there's no net movement or water movement. What it means here is that the amount of water that goes in there, into the red blood cell, is the same amount of water that comes out. And because there's no net movement of water molecules, the cell, the RBC, okay, stays where? Remains
the same. All right, great. Okay, so the RBC remains what? Remains the same. All right, great. Good. So now that we've known these three types, okay, when you put this water in concentrated and stuff like that, okay. Now my question is this: a nurse is in a hurry to go home. Okay, so let's say the nurse. So let me ask you this question. Okay, so a nurse is in a hurry to go home. Okay, and then so you see when you go to hospital, and then they are giving you drip. The drip that they give you is we call it physiological saline. The concentration is similar to that of your blood. Okay, so let's say, so a nurse mistakenly puts the still water as an infusion for a patient instead of what? Instead of physiological saline. Okay, so the drip that they will give it to you and stuff like that, okay, we call it physiological saline. That one, the concentration is similar to that of your blood. Okay, great. But in this case, the nurse was in a hurry to go home. She has closed her duty. She's in a hurry. She's tired. She came overnight. She's tired. And she mistakenly puts the still water, okay, as an infusion for a patient instead of physiological saline. Okay, all right. What will happen to the patient? What will happen to the patients? To the patients. Yes, this is real life scenario. What will happen to the patient? Yes. What will happen? Yes. You should expect this type of questions. So.
Doc.
Yes, please.
When the water, the distilled water that the nurse uses, it means the cells of the patient, that will be the water will be hypertonic solution.
Okay, good.
So therefore the blood cells or the red blood cells of the patient will absorb through more water through osmosis, and therefore the cells will burst.
Excellent. Well done. That's excellent. Okay, good. So the doctor has spoken. Good. Well done. So here, when the patient is giving the still water instead of physiological saline, this the still water is what it is there. So as it's mistakenly like, oh, they picked it, they are similar, the distilled water is there, something they mix the distilled water with some by using hurry. So just put and connect the wrong tube and put the distilled water, okay, mistakenly. Okay. So when it happens like that, what is going to, okay, the water, okay, as more water, as more distilled water, okay. So as more distilled water gets into the blood, okay, the blood will become, okay, as more distilled water, okay, so as more distilled water, okay, gets into the blood, okay, the blood, okay, becomes what? Becomes hypotonic to the red blood cells, okay, and this will cause influx of water into the red blood cells, causing them to become what? Turgid and burst. Okay, so the following day, if the person put a drip on you in the evening, the following day you come and see the patient dead. Okay, the person will suffer from what? Hemorrhage.
Okay, all right. It will lead to the hemolysis. Okay, so it will lead to the hemolysis of what? Hemolysis of the RBC. Okay, red blood cells. Okay, so all the red blood cells will burst. The red blood cells will burst. They become turgid and burst. Because the physiological cell line that we use is of the same. So our blood and the red blood cell, they are in what? They are isotonic. Okay, so that is why nothing happened. But the moment you do that, okay, you cause the word hemolysis. Okay, so they will burst. The cells, the red blood cells will burst, and then the patient will die. Okay, all right, good. Now let's see that of plant cell. Okay, let's see that of plant cell. All right, let's see that of plant cell. Okay, so let's see osmosis in where? Osmosis in where? The plant cells. Okay, great. Now when you come to osmosis in plant cell, okay, so the same approach here, let me pick image because the explanation that I've talked about is the same. So let me just pick image here and then use it to explain. Okay, all right. Okay, so that to move faster. Okay, so let me pick because the explanation is the same. Okay, so let me pick this one. Let me pick an image here and then I'll use it to explain. Okay, because the explanation is the same basically. Okay. Oh, all right. So let's see. If you look at my screen here, okay, great. So great. Now, if you take this water, so you can see that, let me start with the hypotonic. So hypotonic solution, you can see that, okay, do you know what? Let me compress this a little bit so that I can write. Okay, good. So, okay. In the case of plant cells, so here, okay, so when plant cell, okay, is placed in word hypotonic word, hypotonic solution, what is going to happen? So water will move from the environment, okay, in the surrounding into the cell. Okay, so water will get into the cell by the process of what? By the process of osmosis, okay, by the process of what? Osmosis, right? By the process of osmosis. And then, so by the process of osmosis, so what will happen to the cell? The cell will swell, right? So the cell swells. You can see that, look at the vacuum becoming so big. The cell will swell, okay, will swell and become what? Become turgid. Okay, turgid. All right, but after turgidity, it won't burst. Okay, it will never burst. It will never burst due to the what? Due to the it will never burst due to
the cell wall, because of the cell wall.
Presence of the cell wall. Okay, great. So here, after turgidity, the cell doesn't burst. Now, so there's one thing you will see here. What you see something here, let me draw something. The reason I don't, okay. So here you see something like that. Let me draw here. You see something like that. You see some arrows here. Oh. You see some arrows here like this. You see arrow here. Okay, arrow here like that. Okay, arrow here like this. Okay, and these arrows are indicating something we call Tego pressure. Okay, they are indicating Tego pressure. So the pressure is equal in all directions. So the Tego pressure here, okay, on all the walls. Okay, all right, here. So the arrows are indicating what? Tego pressure. So let me add it here. Okay, so these arrows are indicating so the arrows are indicating Tego
Tego pressure. Tego pressure. So you can see the reason it doesn't burst is that, okay, the reason it doesn't burst is that the Tego pressure is equal, okay, in all directions. The Tego pressure is equal in all directions. Okay, it's equal in what? In all direction. All right, good. So the Tego pressure is what? Is equal in all direction. All right, good. So the Tego pressure is pressure due to the what? Due to the wall. So Tego pressure is created as a result of the. So Tego pressure, T pressure is created due to the cell wall. Cell wall. Okay, great. So when you place the plant cell in what hypotonic solution, water will get into the cell by the process of osmosis. It will swell and become turgid, but it will never burst. And the reason it will never burst is due to the presence of the cell wall. So the cell wall creates Tego pressure that is equal in what? All direction. So the Tego pressure here, the Tego pressure here, they are the same, okay, in all directions. So because of the Tego pressure, the cell will not burst. So remember that the Tego pressure in what? In a cell, as a result of osmosis, is created by the what? It's created by the cell. Are you there? I can't see you people. Okay. Are you people there? Yes. So the Tego pressure here, the Tego pressure is due to the what? It's due to the cell wall. Okay, and it is equal in all directions. That's why the cell will not burst. Okay, so it will be more or less some sort of equilibrium. So the cell will not what? Will not burst. Okay, all right, good. So because the Tego pressure here is equal to the Tego pressure there, equal here. All right, good. Okay, so that's the Tego pressure. Right, good. Any headache so far? Any headache so far? You see, I made mention of this Tego pressure because there's sometimes in the books they will tell you what is this arrow indicating, and don't forget that the arrows are indicating what? Tego pressure. Okay, all right, great. Then let me duplicate this slide here. Let me duplicate this slide here, and then let's see for the. So this is for what? The first one that we did is for hypotonic what solution. Now let's see for the isotonic solution. So isotonic, so plant cell in what in isotonic solution. What will happen? There will be what?
Hello, Doc.
Yes, please.
You see, the image that you have highlighted, is it the vacuum that is shrinking, and or is the whole cell? Because I
I've
know
not talked
the
about shrinking. Please let me take a better image. Okay, please let me take a better image. Okay. So it's not only the vacuum that is shrinking, but you see that the water will enter the vacuum. Okay, so it's not only the vacuum that will shrink. Okay, so it's the living component. That's why we use the term. If you remember that what we said the previous one, we use the term word. We use this term. We use protoplasm, and protoplasm is the living component of the cell. So we use a technical word, okay? So that's the protoplasm. So it's not the vacuum alone. The vacuum, the nucleus, everything. But you see, anytime water is going there, it's the vacuum which is more or less it's occupying quite significant space in the cell, in the plant cell. So you see that the water is entering the vacuum. Okay, so you see that like
in the case of the word, this becoming turgid, you can see that the vacuum will collect almost all the water, but the water will enter almost every part. But the vacuum, because vacuum is more or less the space there. Okay, that's why. But the shrinking, if it is shrinking, it is the entire living component. Okay, you know the cell wall is not part of the living component of the cell. The cell wall is not. The cell wall is regarded as non-living. Okay, great.
Okay.
Okay, so here when you come to the word the isotonic solution, in isotonic solution there's what? There's no net movement. Okay, in isotonic solution, okay, in isotonic solution there is no net movement of water molecules, and so the cell remains the same. All right, good. Okay, now let me duplicate that for what? For that's for the what? This is for isotonic. We did this one for isotonic solution, and let's see for the hypertonic solution. Okay, let's see for the hypertonic solution. Okay, so for hypertonic solution. Okay, so for hypertonic solution, what is going to happen? So here you can see that the cell begins to, okay, so initial stage to begin to lose water. Okay, so first of all, okay, so water will exit the cell by the process of what? By process of osmosis. And what will happen? Okay, as it continues, the protoplasm begin to shrink. Okay, so here as it loses water, it's what? It reduces, so it becomes what? Flaccid. So like a flaccid. Okay, I don't even like this image. I have to change it. Okay, let me see. Become flaccid, then it will what? Then to begin to what? Shrink. As the loss of water continue to begin to shrink. Okay, shrink. Okay, good. So as it is shrinking, okay, so you know, let me get a proper image, better image. Okay, so let's say. Let me take a better image. Okay, let's see a better image. One cell in. Okay, I'm not getting better image. Okay, let's see. Let me see Dreamer. The image better. Okay. So let's see. Let me take this one like that. Copy. Okay. And then still it's not the best that I've done. The best that I've done. Let's see. Okay. It's still not the best that I want, but let me take this one like that. Okay. That's the one. Okay, good. Let me. I've gotten this one here. Okay. Okay, great. All right. So something like this. Okay. So here, the cell what? The cell begin to shrink. Okay. All right. Great. Like that. So as it shrinks, you will see that. Okay. As it starts shrinking. Okay. Let me even put. Let me draw. I should draw that. Okay. So let's say. Let me draw here. So if as the cell begins to shrink, so let's say initially the cell was like this, like that. Okay, beautiful with a cell wall, okay, and cell membrane and stuff like that. Then the cell wall will still be intact like that, okay? But so when it starts shrinking, you will see the cell membrane. The cell membrane is part of the living components of the cell. So you see that the cell membrane begins to wall off itself, okay, like that. So you see the cell membrane will begin to wall off itself, okay? So and then the last one, at the last stage, okay, so the cell wall is still intact here. Okay, so cell wall is still intact. So then everything will shrink at the center. Everything will shrink at the center. Okay, so here we have the word. So here the shrinking, so the protoplasm begins to what? Protoplasm begins to become flaccid and shrink. And then the shrinking, okay, so the word. And then so what you see is that the word, the cell membrane walls off from the cell wall, maybe comes off rather, it comes off from the cell wall, okay, and then comes off from the cell wall, and then what is going to happen? Then it will what? Then it will shrink at the center. All right, great. So this process is what we call what? This process is what we call plasmolysis. Okay, so the shrinking of the protoplasm of the plant cell, okay, is known as what? It's known as plasmolysis. Okay, so the equivalent of plasmolysis in animals is called crenation. So, okay, the shrinking of protoplasm in what? In plants, okay, is called plasmolysis, and that of what? That of animal is called crenation. Please, any questions so far? 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 Hello, any questions so
Doc.
far?
Yeah, I want to. There has been instances where you see farm products like tomatoes, garden eggs, sometimes plantain itself. There are instances where you see them bursting. You see that it is bursting.
You see that
the fruit itself is bursting. Like tomatoes,
Yeah.
when it overripes, within a short time it will burst and it will explode. My little mind tells me that it can we classify that as a typical example of what do you call it, plasmolysis. Let me take plantain. The plantain itself, when it grows, or as time goes on, whilst it is growing, the stem is very soft, and with time you see that it will burst and water will be coming out of it. The plantain stem itself. Is it not a typical example of?
No, what we are talking about is in cells. Plantain bursting is basically being overgrown, a lot of food deposited in there, and then too much pressure in it, bursting. It has nothing to do with water going in there. This we are speaking in relation to water going in or water coming out by the process of osmosis. After plantain being overgrown and then bursting is just like continuous deposition of food in it, and then it will burst and then like that. Yeah. So it just continues the position of food that will cause it to burst, but it has nothing to do with what? With water coming out of it. Here, even look at the description. The description was water isn't either exiting or influxing in relation to the concentration of what? Of the solution of the cell content in relation to that of the environment. That's what it means. It has nothing to do with. So the example you said has nothing to do with osmosis. Okay.
Okay.
All right, good. So the next one we're going to talk about is a active transport. Okay. All right, great. Yes, Abdel, your hand is up. Let me hear from you.
Doc.
Yes, please.
Please, I am particularly about that of the question you asked about a nurse infusing distilled water instead of the what?
Physiological saline.
Special physiological saline. In case another nurse or a doctor or any medical official come to find out, but the patient is still alive, what will be the immediate intervention in order to stop the patient from dying?
In this case, let's see what would be the best way. If it is in the blood, if the patient is still alive and even a lot has gone in there, and then the cells are bursting. Let's see what should be. Okay, well, please, unless I think about it. Okay, so maybe I'll give you answer later on. Okay, I'll think about it and give you answer later on. Okay, so but let's go to the active transport. Okay, so that because I have to wait.
Thank you, sir.
So let's see active transport. When you come to active transport, okay, when you come to active transport, okay, so it is the word. It is the, so here we can say that it is the movement of a word. It is the movement of molecules, okay, it is the movement of molecules, all right, from a region of a word. Higher, okay, no, this one's good. I'm coming. From a region of lower concentration, from a region of lower, okay, look at the way I'm going to do for lower concentration, from a region, region of what? Region of lower concentration of solutes to a region of a higher concentration of solutes. Okay, so here solutes. Okay, I've deliberately defined it this way. Solutes. So here, the molecules themselves are the ones that are going to move. They will move from where they are low, okay, to where they are where they are high. And so clearly you can see that they are where the molecules are moving against the concentration gradient, the concentration gradient. Okay, the molecules are moving against the concentration gradient. So we can see that where they were, here the molecules are low. So let's say, so this is reverse of what? Of diffusion. Okay, diffusion, reverse of diffusion. And in here, okay, molecule movement of molecules, you see here, like just like diffusion, but in this case, molecules are moving from where they are low, okay, to where they are high. Okay, all right, and then so the molecules are moving against the concentration gradient. Okay, so when you are moving molecules against concentration gradient, so here So Danny, please, I'm in class. I'll finish very soon and I'll call you. Okay. Okay. Please, I'm in class. I'll finish very soon and call you. Okay. Great. Thank you. Great. So we have so here, true selectively permeable membrane. Okay, so so we have what? So here, good. So true a selectively permeable membrane. So the molecules are moving against the concentration gradient. Okay, and anytime you move molecules against concentration gradient, what happens? You need what? You need energy. So here using energy. Okay, using energy from the cell. All right, good. So here, movement of molecule from a region of low concentration to a region of what? Higher concentration through a selectively permeable membrane using energy in the form of ATP. Okay, using energy from the what? From the cell. Okay, great. And here, the reason energy use is involved is because molecules are moving against the concentration gradient. Molecules are moving against the concentration gradient. Okay, and when molecules are moving against the concentration gradient, what happens? We need what? We need energy. Okay, so for cells, those transports, okay, active transports, okay, active transport tend to occur through what? Through special, so active transport, okay, occurs through special what? Special membrane protein called what? Called pumps. Okay, so pumps. Okay, so special membrane protein called what? Called pumps. Okay, so through special membrane protein called what? Pumps. So when you have the special membrane protein we call what? Pumps, then okay, so these pumps work with what? Using what? Using ATP, using energy. All right, good. So anytime you write, molecules are going to move against the concentration gradient, then energy is what is required. So let's take a instance here whereby we have what? Let's take it this way, that you have glucose here. This is glucose here, and then the concentration is 2 molar here, and then there's another glucose here. The concentration is what? 5 molar here. Okay, and let's say we've built what? We've built permeable membrane, some selectively permeable membrane. You see here, I use the word, look at the term I use. I didn't say semi-permeable membrane, I say what? Selectively permeable membrane. Okay, okay. So let's say I build selective permeable membrane here between the two. And if the 1, 2 here moves up here, okay, if the molecule is different color, if the molecule here move to this one, the glucose here move like that. Okay, okay, so you can see that the concentration is against the concentration gradient. Like if it's along the concentration gradient, we expect the glucose here, the glucose in this side to move this way. But if it's going this way, it will use what? Energy to do it. Okay, so when it uses energy, okay, it will what? It will when it uses energy, okay, to pump, okay, the one here to this side, that is active transport. Okay, your wisdom, your hand is up, please let me hear from you.
Yeah, so I just want to know the difference between the selectively permeable membrane and semi-permeable, because you emphasize it. Yeah.
Selective permeable membrane, we use it exclusively for water.
We use it exclusively for water.
We use semi-permeable membrane. We use semi-permeable membrane exclusively for water.
Okay.
And then we use selective permeable membrane as generic. So it is selective because when you say selective, it doesn't allow all molecules to cross.
Mm-hmm.
So it will allow certain molecules to cross where other molecules will not be allowed to cross. So that's why it's selectively permeable. Okay.
Okay. But semi-permeable is also selective in terms of
it's selective, but it's exclusively to water. So when we are using water, we use the semi-permeable membrane here.
Okay.
So here we have two mole here, 5 mole here, and then the glucose, okay, is still being moved across the cell membrane like that. Then it will require energy to move this glucose. Okay, so when it requires energy to move the glucose, then it is what this particular process is what active transport. Why? Because the molecules are moving against the concentration gradient, and against concentration gradient means that it requires what? Energy. Okay, great. All right, good. So let's see. So which means that the cells that are going to involve in what? Active transport. What is the obvious organelle that you are likely to find there? So cells that are going to be in active transport, what organelles do you think would be quite dominant there? If I may ask. So cells.
Yeah,
mitochondria.
Cool. So cells that are involved in what? That are involved in what? In active transport of molecules. Okay, molecules have what? Numerous mitochondria. Okay, mitochondria. Okay, so produce energy. Okay, so mitochondria to produce what? Energy. All right, great, good, well done. Now, so let's see.
No.
Yes, please. Please go ahead.
Can you call this one? Yeah, can you call this process as a facilitated one?
It's not facilitated. Facilitated doesn't require energy. Okay, facilitated diffusion. Facilitated diffusion, it doesn't require energy because remember molecules are moving along concentration gradient, and that one doesn't use pump. It uses channel.
Okay.
Okay, it doesn't use pump.
Okay,
Pump is like another name for pump is called even ATPs. Another name for pump is called let me even put it here. Another name for pump is called ATPs because it uses what? ATP. So another name for pump. Okay, those are pump is called. So for facilitated diffusion, that one it uses pump and it doesn't require energy. Okay, because molecules move from higher to lower. But here molecules are moving from lower to higher. All right, good. So let's see the factors that are affected. So factors that are affected what? That affect active transport. Okay, yeah, so let's see factors affecting active transport. Yes, what are the factors affecting active transport? Yes. Please, what are the factors affecting active transport? Yes. What are the factors affecting active transport? Yes. Yes, what are the factors affecting active transport? Yes. Factors affecting active transport? Yes. Factors affecting active transport. Yes. Factors affecting active transport. Yes. Good.
So can you talk about temperature?
Temperature. Good. So ultimately, the first one is the concentration gradient. Okay, so I won't elaborate on this because I've talked extensively about it. 2 is what? Temperature. Okay, temperature. Okay, so 2 is temperature. Okay, so 2 is temperature. All right, 2 is what? Temperature. All right, good. And then what else? So here we can even talk about the size of the molecule. Okay, size of the molecule and play a role. Size of the molecule. Okay, the size of the molecules. And then 4, we can also talk about what? Energy availability. All right, so energy availability.
For energy, can you make it ATP availability?
Okay, energy. The energy is not always ATP though, but this is basic, so I don't want to talk about that. There could be something for secondary active transport. It's not always ATP. Something can use electrochemical process to do that. Okay, it's not always ATP. All right, good. So energy availability. Okay, good. So these are the factors that affect what the affecting the rate of what the rate of active transport. And let's see the significance. So significance of what active transport. Okay, active transport. Yes. One. Significance of active transport. Okay. Yes. Give me one. Yes, please. Okay, please. I'm listening. Yes. Okay. So one, we can talk of what absorption. Okay, so implant, we can talk of our, so let's say implants. So you can talk of what absorption of some mineral salts by the roots of plants. Okay, so that's what I said earlier on. So absorption of what? Of mineral salts, some mineral salts by the roots of plants. Okay, so sometimes some of those minerals, so they're even still higher in the plant than in the soil. But plants will use energy to pump them because they are those nutrients are very, very important. So some absorption of some mineral salt by the roots of what? By the roots of plants, okay? By the roots of plants, okay, good, can occur by active transport, okay, especially if the concentration is higher in plants than in the, okay, good. Then we have the two, okay, we can talk of what, we can talk of absorption, okay, absorption of what, of glucose, okay, and what, amino acid, okay, acid by the villi of the small intestine, by the villi of the ileum.
Okay. Look, I think we are looking at plants. Oh.
The word. Okay, so I think the plant, that is all that we can. So okay, so let's see animals, you can put animals on top here. Good. So all right, so by the villi of the small intestines, okay. By the villi of the small intestines, okay, good. And then another one for animals, you can talk of the reabsorption, okay, reabsorption of what? Of some nutrients, okay, nutrients in the what? In the tubules of the kidney nephron, okay, nephron. Even when the concentrations are, even when we have enough of it in the blood, like glucose, even when we have enough even in the blood, still they will use what? The body will use ATP to pump the glucose into the blood. Okay, you will still use the ATP to pump. So you don't expect a normal person, like a healthy person, to pee with glucose in the blood, in your urine. Like when you pee with glucose in your urine, it means you are suffering from diabetes mellitus. Okay, so we don't expect it because no matter what, if they don't reabsorb it by diffusion, they reabsorb it by what? By active transport. Okay, all right. So just to make sure that everything goes, because glucose is very, very important. So because it will be reabsorbed into your body, because the excess glucose, when you have even excess glucose in your body, it wouldn't even come to your urine. It will still be stored in the form of what? Glycogen in your body. So it wouldn't pass your urine. So even when you have enough glucose in your body, the body will still reabsorb the glucose in your blood and store it in the form of glycogen, so in the liver, so that anytime you need it, the body will make use of, they will convert the glycogen into glucose for you to use it. Okay, So when you don't have enough of it, okay, when we don't have enough of it in the blood, it will be reabsorbed by the process of diffusion. When you have enough, or there's even high concentration of glucose in your blood, still there will be what? There will be reabsorption. Okay, there will be reabsorption. They will use energy to reabsorb either the proximal convoluted tubule or the distal convoluted tubule. They reabsorb the glucose there by the process of what? Active transport. Okay, into your blood. Okay, all right, good. So this is how far we can go so far as this one is concerned. The last part of the day is the bulk transport. Bulk transport. Okay, the bulk transport. Okay, the bulk transport. Okay, so the bulk transport, at last time I gave you the head start of the bulk transport. The bulk transport is a special form of active transport because this one, it requires energy. But just that the bulk transport is such a way that the molecules are so big that they can't go through the wall. So they need to be carried in a vesicle. Okay, so let me show you an image of it. Okay, so let me show you an image of it. Okay, let's say bulk transport, let's say isocytosis. Okay, let me show you an image of it. Okay, good. Okay, so let's see something like this. Okay, okay, so let me show you something like this. Okay, this one is too small. Okay, I'm going to show you a picture right now. Okay, great. So copy. Okay, and then put it here. Okay, so let me put this one here. Okay, then let me do another one. Let me take another one here. Okay, so okay, let me take another one here. All right. Okay, it's still loading. All right. Now, if you look at, oh, where is it? The one I brought. Okay, let's see. Okay. So, okay, so there's another one here. Let me copy this one as well. Okay. Oh, maybe this one. The copyright is not allowing us to copy it. So let's see. All right. Okay, you know what? I can even crop it. Let me cut it because the image is nice. Okay, so let me. Okay, so let me take okay. Okay, great. So the images I'm taking, they are not coming. Okay, great. Now you can see from my screen here. Okay, you can see from my screen here. So this is bulk transport. And when you come to bulk transport, there are two things we talk about. Okay, so we talk of the word. We talk about the exocytosis. Okay, and the word, and then and endocytosis. Okay, let me see whether the other image has loaded. Okay, let's see. Copy this image. Okay. Okay, great. So let's see. Okay, so we have, let me see, this one will come. Still, okay, great. Oh, right. The computer screen is frozen, doesn't want to do anything. Oh, why? Okay. All right, good. So, so when you come to what? So we have the what? The isocytosis. So when the molecules are big, and so big molecules like that, they can't actually go through the what? They can't actually be going through by diffusion or the normal active transport. Okay, so we have two main types. We have the exocytosis and what? And endocytosis. Okay, so we have the word exocytosis and what? And the endocytosis. Okay, and so for endocytosis, what happens is that we have the word, we have the, okay, we have the, okay, so with the exocytosis, we have the molecules coming out of the cell, and then endocytosis, we have the molecules where we have the molecules getting into the cell. Okay, so if you look at my picture on the screen, okay, so you can see my picture on the screen that, okay, look at the exocytosis. Okay, if you look at the exocytosis and endocytosis, the exocytosis is something coming out of the cell. Okay, all right. So, like for instance, the exocytosis, we have the like during the transmission of nerve impulse. You see, at the neurotransmitter. And then let me get you this picture. At the neurotransmitter. Okay, so let's say neurotransmitter. So let's say at the synapse. Okay, so neurotransmitter. Okay. Okay, good. So let's say if I copy this image for you. Okay, great. Copy this image. Okay, good. So let's see something like this. Okay, let's see if you look at this one. Okay, great. If you look on my screen, okay, so you can see that at the neurotransmitter, okay, when during the transmission of nerve impulse, okay, during the transmission of nerve impulse, you can see that at the synapse, okay, the neurotransmitter will do what? The neurotransmitter will be released at the what? At the synapse, okay, all right. So the neurotransmitter is quite a heavy molecule. And because it is a heavy molecule, it's not going to what? Diffuse. It's not going by diffusion or the normal active transport because the molecules is very huge. So it is going to what? It's going to form, to be in a vesicle form and then to be released into the what? Into the synapse, okay? Great. So if you look at this two here, we have what? We have the exocytosis. Exocytosis is when something is coming like the enzymes, milk, okay, milk coming out of the lactocytes in the breast, okay, neurotransmitter coming out of the what? The axon, okay, and into the synapse. All right, great. So those ones are what? Exocytosis. And endocytosis is something coming into the cell, okay, all right. And then this something coming into the cell, we can group it into two. For the endocytosis, we can group it into two. Pick one because some of you have to join some chemistry class, so we need to hurry up. Okay, good. So when you come to the endocytosis, we can get it in what? Into two. Okay, we can get it into two. Hello.
Some of us have lectures around.
Yeah, that's why I'm actually ending. I'm actually ending it right now. I'm just saying one, two, three, and then I'm ending the class. Okay, so great. So when you come to the endocytosis, the endocytosis, okay, endocytosis is basically something coming into the cell. So in this case, we have two types, okay? For the endocytosis, we have two types here. Okay, so let's see, let me duplicate this slide. Okay, great. All right, great. So let's say for the endocytosis here, okay, we have two types. Okay, for endocytosis, we have two types. When it is what is going here into the cell, if it is liquid, okay, if it is liquid, we call it what? It is pinocytosis. That is when it is liquid. And when it is what? Solid, it is what we call it what? Phagocytosis. Cytosis. Okay, so for this one, so phago what? So that's a my screen. Okay, that's a phagocytosis, and then what? Phagocytosis. Okay, great. All right, good. So when you talk of the pinocytosis, you can say I'm just moving the screen up. This thing is frozen. Okay, great. Let me. Okay. All right. So here, so for this is what for liquid coming into the world, liquid coming into the cell. Okay, and then the phagocytosis is what? It's a okay. That's a solid. Okay, solid. Okay, good. All right, good. That's a phagocytosis. So endocytosis is basically something coming to the cell. Okay, good. All right, and then they are of two types. When it is liquid, it is pinocytosis, and when it is solid, it is phagocytosis. Okay, and these are bulk, they are heavy. And as a matter of fact, this bulk transport, they require energy. Actually, they require energy. So they are actually form of what? Active transport. Just that they don't go across the cell membrane, just like the way diffusion and normal active transport occurs. Okay. So they will go in the what? If you look at the picture here, you can see that the vesicles. Okay, the vesicles are what? They are in the form of what? Okay, they are in the form of what? Of a vesicle. Okay, and because they are in the form of this vesicle, okay, all right, so they will vesicle like that, and then it will come out of the cell, or it will get into the cell, because the molecule is so big that it wouldn't be able to what? Diffuse or go by normal active transport. Okay, but this bulk transport requires some sort of what? So they require some sort of what? Energy. Okay, so they both exocytosis and what? Okay, so bulk transport requires what? Energy as well. Okay, so the movement of to move the vesicles across requires what? Energy. All right, good. So this is where we'll end the class, because the chemistry people need to go. Okay, so I'm going to save what we've done here, and then I will send it to you. Okay, so we need to get one more meeting. What time would you suggest? We still have what time would you suggest so that we do the DNA and genetics? What time, please, would you suggest?
Sir, we can use next weekend too.
Next weekend is when you are going for your practicals.
Practicals, yeah. Since you're not having practicals in this block,
Sir,
you can use.
Okay, we won't have
the evening time.
Chemistry people have.
The chemistry people have practicals. Evening today.
Yes, sir.
Hello. Evening today.
Yes, sir.
Hello. Okay. Evening time. Would that be okay don't other people not having class today? Okay. So evening time 6 o'clock. Okay, that will be let's see, Saturday. Okay. Well, I think I will squeeze time for you. Okay, evening time is fine.
Evening today 6 to 9. Yes, sir. No.
So,
sir, not tonight.
That's why.
Not tonight. Next evening.
No, next Tuesday to get close to exams. It's getting too close to exams.
Sir, this evening, you support this evening.
Not today,
all right.
Evening. Sir, please, they are lying.
Evening.
Not today.
Evening.
Why not today?
Not today. Because
today.
This evening will not
Why
help us.
this evening will not help you? Let's say it so that we know. Because listen, sir, the thing is that
where are you going?
Listen. Hello.
The chemistry we have class from 12 to 4.
Yes, 12 to 4. So why don't you
so from 12 to 4 you close? You have 2 hours rest. Then we'll continue at 6 o'clock.
And then we'll continue. Because listen. We are not
At
so
that
sure
time,
what.
most of us have also not closed from work. Oh.
Hello.
Some of us will be writing our
Hey,
lesson notes for the week. Oh. Some of us will be
please
writing our lesson notes.
listen. Hello.
Do you still write lesson notes?
Oh,
And also some of us will be cooking.
Why don't you write lesson notes?
listen.
We do.
And
Okay.
your lesson
Why
notes,
so
you still write
sensitive?
lesson notes.
I will leave you.
Oh, allow us to talk.
One after the
Allow
other,
doctor to
after
talk.
the other.
Listen, one person to talk.
Silence,
One after
silence.
the
Sir,
other.
please, you can talk. I'm your team.
Please, folks. Me sacrificing that time is purely sacrificial.
It's true. It's true.
Oh, you guys are not aware. The extra meeting
Sir,
that I need you.
sir,
Listen, the extra time that I need you is purely sacrifice. It has
sir,
nothing to do with God. God doesn't pay me for that.
sir.
It's a sacrifice. So it's a favor. It's a favor. Maybe you guys are not aware. It's a favor.
Yes, sir, but you want that favor to be like tomorrow or
I
tomorrow
think
next, for
the best
example,
option
Monday.
is for you to stipulate the time you are going to meet us.
So, no, because you guys, everybody here is a grown up. That's why we want to discuss. I don't want to impose anything on anybody. So I'm thinking that today, this evening, okay, this evening from 6:30 maybe to 8:30, that's fine. Where I will end there, I've set my end of questions. Please listen, let me land.
Okay, 6:30 to 8:30 is done.
If you come, it's up to you, okay? But even God in heaven knows that I've done more than necessary because most of it twice the time. So it's up to you if you're able to come. Okay, wherever I'm going to land today, that is where I need to meet you now and finish we've not even finished the cell cycle. We've not gone the details of mitosis, the DNA thing, and then the genetic parts. Okay, good. We need to finish, so we need to cover those aspects today, and then God willing, next week we'll use it for solely Mendelian genetics. Okay.
Bam.
I've said the same questions already. If you don't come, then we'll finish. Please. I don't think I'm a very busy person. I'm only sacrificing time, squeezing time, and canceling meeting here and there to meet you. Okay? Not that I'm so relaxed. Like no, I'm not. I'm very busy. Okay, so it is up to you. So this evening, 6:30, we are meeting. Okay, I'll go somewhere. I'm going for some stuff. I'll run away from there. Okay, and then I'll come by 6:30. I'll come here. I'll rush to the office, and then I'll come and finish with you. Okay. I wish you all the best. Have a good day, and God bless you. Thank you.
Doc, please, that's live.
Thank you.
Yes. Why don't we finish all this evening so that I will top everything up this evening, and then I'll send everything together.
Well noted. Well noted.
Okay, sir.
Well noted.
Thank you. So 6:30 this evening we are meeting. Thank you.
6:30. Well noted. God bless you. Thank you very much.
Thank you.
Final say, if you like, talk.
Hello, sir.
Yep.
Please, are you going to give us examinable topics since the stress are many?
You're supposed to. I've given you people examinable topic. I've given you. I've sent some plenty questions to you on cytology and genetics. There's nothing like examinable topical. Everything I'm teaching is important. It will come. You'll be examining everything that I'm teaching. Everything I'm teaching, I'll set questions to cover everything I'm teaching. Okay? Everything I'm teaching. Okay? It will cover everything. So you see your quiz question first, but the quiz like that, maybe I will not ask so much of plenty genetics because by that we've not covered there for the quiz. But for the end of term,
You promise that you will be fine.
yes, you'll be fine. So yeah, but I'll set standard questions. My questions are always standard. Okay, so you'll be fine. Okay, God will be with you for sure. All right. Hey, that thing is hot, so you people shouldn't disturb daddy. Yeah.
So can we have 5 questions?
Well, the question that I sent you, the trial question I sent you, is far better than past questions.
Oh, okay.
The trial question that I've sent you, have you gone through them?
We are still going through.
Yeah, so go through the
Hello.
past question. That's one I sent you. That's far back. Because that one is more or less the question that are like that's similar questions that I've set. And I've given a set of them to you.
Okay.
So that one is far better than past question. Because past question, I'll put the previous question and place them side by side and make sure I don't repeat questions. Okay? I
Hey,
won't
dog.
repeat any question. Yes. No, I don't do past go here. So learn everything that I will teach you. Okay.
But the
All
first
right.
chance,
Thank you.
the first
You'll be
chance,
examining.
you will not get it. The soil
Yes,
science,
there were.
you get the question. We have not read that.
The soil science. The soil science I've been giving you a lot of trial questions. The true of us, plenty stuff like that. The questions that will come, they are not too different from what I've been giving to you.
Doc, we wanted something like the way you gave for the cytology, so that
It's
we'll read
time.
on that one
It's a
too.
lot of time. I'll try, but I'm very busy now as well.
Doc,
It takes a lot
try.
of time to.
We beg.
Oh, I'm very
For the
busy.
sake
I'm
of
very busy.
our agenda, try.
I will try, but I'm extremely busy. Extremely busy. I always squeeze time to it.
Thank you.
All right. So thank you so much. Okay, have a good day. Okay.
Same to you, Doc. God bless you.